am so pleased to welcome you to this year's Nix Lecture. For those of you who haven't met me yet, my name is Sarah Heilsorn. I have the distinct privilege of being the chair of the department following Alberto Soleillo's stepping down last July. This annual event is a way to bring together and connect our Stanford material science and engineering community. And those connections are meant to bridge across the generations. And it's been a lot of fun already this morning seeing our current students and postdocs interacting with our alumni and seeing our alumni reconnect. We have an invigorating schedule planned for you, and I'm really looking forward to hearing all of today's speakers. Whether you're an alumnus from our undergraduate program, which as many of you were telling me today, and I know it's been an on-again, off-again undergraduate program, from our master's program, our PhD program, or a postdoctoral scholar, we welcome all of you. Last year, the Stanford School of Engineering celebrated its centennial, marking 100 years of a coordinated strategic approach to engineering education and research. To commemorate that milestone, we've shared with each of you a notebook for the School of Engineering Centennial. Of course, though, as many of you know, our department reached the centennial landmark before the school, right? So we celebrated our centennial some years ago in 2019 because the predecessor to our department, which was the Department of Mining and Metallurgy, was founded 5 years before the School of Engineering. Since our founding and through the evolution of our department to the way it is today, we've really held two foundational pillars that guide much of our decision making. The first, pursue excellence. And the second, to nurture and support the individuals in our community. I wanted to share just a little bit about the current department and how decisions we're making now illustrate that those two founding principles are still very much guideposts for us. First, if you're going to pursue excellence, you have to recruit excellent individuals. And that means bringing people with the best ideas from everywhere around the world together so that we can learn from one another and push one another. At this time of national public dialogue about the value of higher education, about the role of immigrants in our community, I was heartened to see that the desire to pursue a Stanford material science and engineering degree is still exceptionally strong across the globe. This year, we had a record number of applicants to our graduate program. And we just finished the painstaking process of evaluating those applications and admitting a full class of outstanding students that we can't wait to welcome to campus next fall. We're also conducting a junior faculty search this year to add a new research group to our department. And similarly there, we received a record number of over 500 applicants for our open position. Together, these two record-setting statistics, I think, are evidence that our world-class reputation for doing the very best research and education in material science and engineering is still very much needed. Our second guiding principle is that our pursuit of excellence is strengthened when we support one another as individuals in our community. Last night, I had the opportunity to share dinner with some of our alumni and speakers, and it was wonderful to hear their personal stories about the inspiring and sometimes quirky individuals that have made our department great throughout the years. I invite you all to take the opportunity today to reconnect with old friends and also to make new friends. This day is about building those connections, and I especially encourage our current students and postdocs to introduce themselves to our alumni and our guests, and to share their current research interests and their career aspirations. I'd like to take a moment and ask, this year we have student and postdoc table hosts. I'd like to ask those who are here who volunteered to quickly stand up so we can recognize and thank you. Please feel free during the break. You might even switch tables, mix it up a little bit, meet someone new. Later today during the lunch hour, we'll also have student-presented posters and, new for the first time this year, alumni-hosted tables, and that will be in the room directly across from the entry lobby during the lunch hour. With that, I'm delighted to introduce our first speaker for today, Jennifer Dione. In addition to her role as a faculty member in the Material Science and Engineering Department, she's also a courtesy faculty member in radiology and a Chan Zuckerberg BioHub investigator and deputy director of a Department of Energy National Quantum Initiative and co-founder of Pumpkin Seed, a company developing quantum sensors to understand and optimize the immune system. She has boundless energy. Our materials science community is also extremely grateful to Jen for her recent service, which she has since stepped down from, as Stanford's inaugural vice provost for shared facilities. Those of us in the materials science community know how critical shared research infrastructure is to the work that we do, and she really helped to galvanize campus-wide support for the modernization of our shared research facilities, and that benefits not just our department, but the whole campus. Please join me in welcoming Jen to the stage.
All right, good morning, everyone. Such an honor to be here, and this morning I was trying to pull together pictures of our department and me and Bill Nix. I just want to give a round of applause to Bill Nix, the namesake of this lecture, the person who's brought us all together and been such a huge inspiration for me and I think for many of us. So thank you, Bill, for all you've done for material science. So we're a little bit behind schedule, but hopefully I can still have my full 15 minutes, but I will speak quickly as I always do. I wanted to share with you just a snapshot of what our lab is doing. But of course, we're all here in a material science colloquium, and I'm sure the main thing on our mind is AI. AI is all the buzz, and for me, and I think also for Reiner, who may be here, our interest in AI really began with poetry. So Reiner and I used to exchange limericks when ChatGPT first came out. And now if you were to go to your favorite AI and ask it to write a poem that suits my mood, we all know this may not be the best prompt. It's somewhat incomplete, right? It doesn't know if I'm excited to be giving this talk, honored to be here with Bill Nix, feeling festive for the Lunar New Year. It needs more information, and it's going to query me for more information. And I think this is a really excellent example of why it's very challenging to develop AI models when it comes to biology. And our lab focuses a lot on developing novel materials and novel methods for biology and biochemistry. For example, if we were to go to our favorite AI and ask it questions like, why is my prescription no longer working for me? Or what's my actual risk of heart disease today? Or how does this new diet improve my stamina? The AI is going to ask for more information, and we actually would not be able to give it the proper information to complete the prompt. And that's because the functional data that matters is extremely challenging to access. So one of the hypotheses that our lab has and why we're working on new materials and new methods for biology is that AI can really only revolutionize medicine and drug discovery if it's equipped with the key functional data that truly drives biology. And I think in many ways, biology has a measurement problem and a materials problem. And that's kind of twofold. On the one hand, there aren't really good models of biology. We've probably all heard of AlphaFold. That's the best model that's out there. It's a revolutionary one. It's trained on a very specific problem. But for all other problems, there simply is not enough data to be able to train good models. And then the second challenge is, even with an internet's worth of data, or abundant data, we still don't have the right way of prompting the AI and giving it the information that it needs to be able to answer some of the dynamic and temporally changing questions about biology that I just put on the previous slide. And if you think about the landscape of biology, and Sarah mentioned that my home department is material science, but I do have a courtesy appointment in the School of Medicine, I've worked very closely with clinicians in my 16 or so years here at Stanford, and they have abundant data about the genome. And all of the cells in our body have the same DNA, whether it's a liver cell, an eye cell, it's all the same DNA. So we know that is static. That isn't changing. And yet, as we age or we mature, our bodies are changing. And that's because the functional molecules of biology, like your transcriptome, your proteome, your metabolome, your lipidome, those are all changing. And those do not have a one-to-one correspondence with your genome. So just as one example, you have 20,000 protein coding genes. And just for one protein coding gene, there are hundreds to thousands of possible proteins that can result. So it's hard to make predictions about these functional molecules of biology. And this is what my lab is trying to address by developing new materials and new methods that allow us to get the necessary data to be able to train better models about biology and get better insights about biology in a high throughput, easy to access manner. And the way that we've decided to do this and really commit ourselves to is based on label-free spectroscopy. So I'll bring you all back to your Material Science 101 class. We use Raman spectroscopy. It is an old technique, over a century old, but a really beautiful method where you send in a laser or a monochromatic light source, and then you simply look at the elastically or inelastically scattered photons, basically the photons that are redshifted. So from your intuition, you send a laser onto a material. Most of the light gets reflected back at the same color, but actually a very small fraction of the photons changes color in response to how the photons have interacted with the molecular vibrations of the material that you're looking at. So it turns out Raman spectroscopy is an extremely specific method. It will tell you precisely your material's composition. The challenge is it's extremely insensitive. So only 1 in a million or 1 in 100 million photons is inelastically scattered. So part of the challenge is, how do you develop materials that can enhance the efficiency of Raman scattering so that way you can get it in a meaningful time period, especially when you're looking at very complex samples and just want to look at individual molecules or individual cells. So that's part of what I'll address today. The second challenge is, how do you interpret that Raman spectra, right? Because Raman spectroscopy, very information rich. Historically, it's been an expert spectroscopist just sitting and staring at the spectra. But it turns out AI has been really transformative in terms of giving us the tools to be able to analyze the different peaks in the Raman spectra and be able to correlate that to the underlying phenotypic signatures that are correlating the spectra to biological function. So some of the materials that we use to enhance Raman spectroscopy are shown here, and since I only have about 15 minutes, I don't want to go into all of the science, but from a high level, you can imagine the experimental platform essentially being a microscope slide with nano patterns, semiconductors, sometimes metals on top. And then what we do is simply image and take spectra of our sample in the far field. And we design those nanostructures in a very precise way such that they can take visible or near-infrared light and essentially shrink it down. It's like a Honey, I Shrunk the Kids. So we're figuring out how to take light that has wavelengths, visible wavelengths of a few hundred nanometers, shrink that down to molecular scale, so less than a few, 20 or so nanometers, and then also guide that light to the far field so it's scattering like a dipole. So it's kind of like we're steering the light in the far field in addition to concentrating it. And here's both a scanning electron micrograph, an optical micrograph, and then also a simulation of how these structures look and how they work. So we illuminate from normal incidence. We have some of these structures. In this case, they're patterned as silicon bow tie structures. And then they take that normally incident free space light, they concentrate it down to that tiny little, like 20 nanometer region that you can see where the electric field of light gets enhanced by over 100x. But the interesting thing, and some of you, or many of you are material scientists, is that because we're working here with silicon in the near infrared, it's non-absorptive, right? So there's no heating, right? Because we're not heating up the material, we're just concentrating light. So that means we can actually interrogate biological samples without destroying them. And then hopefully this video plays, but here's just kind of an example of the sort of data we collect, where you can see about 300 sensors lighting up here. These are all silicon, so there's no fluorophores, but we're just putting our biological sample on top. And from the pattern of how those resonators are lighting up at different wavelengths, we can infer and interpret what biological molecule or cell might be on those sensors. And you saw the scale bar. I know Bob Sinclair is in the audience, so he taught us on day one, always include the scale bar. But then just to hone in on if you now pattern these over a large area, you can fit millions of sensors per square centimeter. So one of my students who might be here in the audience, Varun Dalia, actually went in response to reviewer number 3 and showed that he could pattern these over a centimeter square area. Hopefully you can kind of see some of the iridescent purple on the screen up there. But in this case, we have 3 to 5 million sensors. For some of you who do biology and maybe think about well plates, this is kind of the equivalent to having over 31,096 well plates, which normally would take up the equivalent of basically like a basketball court. But we're able to get these extremely strong electromagnetic fields in very small volumes, which is proportional basically to what we call the quality factor, so high Q. And then, since I mentioned Raman spectroscopy, this is probably one of the more detailed plots I have, but all you have to do is take away the fact that if you are not illuminating on resonance, or if you don't come in with the right polarization of light, you're not going to get any signal, right? Because Raman spectroscopy is so weak. But we engineer these nano antennas in such a way where they can strongly amplify or enhance that Raman spectroscopy signal. And then when we look at different sensors, in this case, all different sensors, but all with the same molecule, you can see basically we're getting that same Raman enhancement. And then what I think is especially exciting, and one of the more recent advances in my lab, is that we can actually do molecular dynamics and first principles calculations like density functional theory to predict the Raman spectra. So we can compute the Raman spectra. And you can see here the theory at the bottom agrees quite well with the experiment. What we do is molecular dynamics to figure out the conformation of the molecule or the macromolecule. And then we take those different conformations, we feed them into density functional theory, which, by the way, is computationally expensive, but with advances in quantum compute, it's actually becoming much faster. And then, based on the Raman spectra and the amount of time that the molecule spends in different conformations, we're integrating for on the order of tens of milliseconds, up to about a second. We can take weighted averages of the Raman spectra based on how long it spends in those conformations and precisely get back the experimental Raman spectra that we predict. So this, for me, is pretty exciting because it means there's actually a path to get information not only about what molecules are there, but what their structure is, potentially in even a dynamic environment and with single molecule resolution. So to summarize the physics, since some of you are not only material scientists but physicists, I like to think about kind of the two different axes being, on the one hand, how long does light stay in your resonator? How many optical cycles will light want to oscillate in your resonator for? And then to what extent can you take that light that's circulating in the resonator and where your molecule is or your cell is, and then send that up to the detector? And historically, there's been kind of a trade-off between these, and maybe some of you have heard about photonic crystals or ring resonators. Those are really great at storing light for extremely long periods of time. but they scatter light in all directions, and it's actually really challenging to get that one-to-one correspondence between what's happening where your molecule or your cell is and what you're actually seeing in the far field, like in a microscope camera. So we basically kind of bring the best of all these worlds together, where if you think about me resonators, those scatter like dipoles, but they're not very sensitive. But we've basically taken principles from nanophotonics, plasmonics, and then incorporated in those high quality factors. And then I think one of the most exciting things about this particular set of structures that my lab has been working on is that they're CMOS compatible. So generally, photonic materials are not the same sorts of materials that you would use for CMOS electronics that you have in your laptops or your cell phones. But we've been able to design materials that are wholly silicon compatible, such that now you can make these sorts of substrates in a manufacturable way and on a large scale, like in 12-inch wafers. So I want to give you two examples of how we're using these to address biological problems, one of which is on kind of de novo molecular sequencing. And probably all of you have heard of DNA sequencing. In this talk, I'll discuss how we're working on protein and peptide sequencing, and this has now been spun out by a pumpkin seed, a company that Sarah mentioned. And then I'll also share with you how we're using these sorts of platforms to predict patient immune responses, especially for immunotherapy outcomes. So on the one hand, we'll look at molecules. On the other hand, we'll look at cells. And I want to be respectful of time, so I'll discuss this pretty quickly, but at least you'll get a flavor of what we're doing, and then please come to the poster session. So for protein and peptide sequencing, those proteins are basically the working molecules of biology. But like I said, it's not a one-to-one correspondence between your genome and your proteome. And there's a vast array of letters. You have 20 amino acids, dozens of post-translational modifications. So it's very challenging to develop methods like DNA sequencing that rely on fluorophores that can capture the complexity of the proteome. So what we do is we essentially take proteins, they're eluted off of cells, and then we put them onto our chip. They're oriented in such a way where all proteins have a carbon-rich terminus, so we orient them by their C-terminus. And then we use kind of an old-fashioned chemistry called Edman degradation, where you come in with this ring-type molecule, it binds to the N-terminus, and then if you do different pH cycles, it basically cleaves off one amino acid at a time. And we've basically engineered those Edman reagents in such a way where they infuse Raman peaks into the protein or peptide spectra that are very specific for which N-terminal amino acid it's bound to. So we have cyclic chemistry. And again, using some of those MD and DFT calculations that my lab worked on, we've been able to engineer these Edman reagents where they still do the cleaving, but they have Raman cross-sections that are extremely bright compared to any of the native amino acids. And then in conjunction with the chip that gives an 8 order of magnitude Raman enhancement, we can do high-throughput single-molecule Raman spectroscopy and protein sequencing. And here just shows that one Edman reagent, what I'm doing is taking the Raman spectra of a peptide with different terminal amino acids, shown here, and then projecting that spectra onto a two-dimensional plane. You can think about this as a PCA, but it's basically a UMAP in this case. And then with the spin-out company, I have Pumpkin Seed. One of the founders and my former student, Jack Hu, has been working very closely with Genentech, where they're interested in using this platform to reveal what broadly would be called the dark proteome. But in particular, they're interested in understanding immunopeptides. These are short protein fragments that are expressed on every nucleated cell and serve as beacons to your immune system as to whether a cell is healthy or diseased. And it's actually very challenging to figure out those peptide sequences with state-of-the-art tools like mass spectrometry, especially when you're looking at an individual patient sample, because mass spec ionizes your sample and you lose over 99% of the information in there. And when it comes to looking at the differences between healthy peptides that all of our cells have versus mutated ones, like in cancer, there's only one amino acid or one letter that's different, so it's extremely challenging to get that information. But here you can just see projections of some of the Raman spectra between healthy and mutated peptides. And then this company, Pumpkin Seed, is working with Genentech basically to sequence the immunopeptides that are on individual patient samples to basically inform next-generation immunotherapies, for example, vaccines against metastatic cancer, where you essentially put the mutated peptide sequences into, say, an mRNA therapeutic or a peptide therapy that if you have cancer the first time, usually you do fine, you remove the tumor, you resect it, but then you can actually prevent the metastatic disease. And there are companies like Genentech, also BioNTech, and others who have had really promising trials using kind of standard tools like mass spec, and we're hoping to improve the outcome of those trials. Okay, so just in the final 3 minutes, I also want to share how we're moving on from molecules to cells. Molecules are kind of challenging because they have smaller Raman cross-sections. Cells, they're bigger, so they have bigger Raman cross-sections, but they're more challenging in other ways because it's kind of like a water balloon of a whole bunch of different molecules. So how can you take the Raman spectra that contains information about the nucleic acids, the lipids, the proteins, and actually provide interpretability to that spectra? So a handful of years ago, my lab started doing single-cell Raman spectroscopy and just wanted to know, could we get Raman spectra plus machine learning models to give us accuracy that compared with state-of-the-art mass spec methods or transcriptomic methods? And here's just an example showing some of the early work we did looking at bacteria. And indeed, it turns out you can actually classify different cell species and strains based on the Raman spectra with an accuracy that is as good, if not better, than mass spec. And the neat thing is that Raman is not just a black box. So you can take your spectra, and then you can computationally perturb it and figure out when does the model accuracy drop down. Basically, what wave numbers correspond to really important features in your classification. And then the neat thing is from the literature and from other experiments that we've done, where you see the accuracy drop down, you know those bands are important, and then you can figure out what those bands correspond to. So that actually tells you about new pathways that might be occurring in biological systems. So here's a project that my student Baba worked on, and now we have Liam also working on a related project where he was looking at the antibiotic resistance of tuberculosis and trying to predict that more rapidly in patient sputum samples. And not only did some of the pathways that he found that corresponded to high accuracy prediction correspond to what's known to be a pathway for resistance to antibiotics, but the cool thing is once you know what those bands are, then you have significantly reduced constraints on the hardware. So he was able to build essentially a field portable Raman microscope and spectrometer in collaboration with Manu Prakash, who's now working to deploy this in TB endemic regions. And then finally, I mentioned that we wanted to apply this to immunotherapy outcomes. We've been collaborating very closely with Amanda Karane and Crystal Mackel and others in the School of Medicine. And with immunotherapy, which has revolutionized the state of care for patients, right now there are no predictive biomarkers of outcomes. So my collaborator, Amanda Karane, is a surgeon. She treats melanoma, and she would love to be able to know, hey, if I remove a tumor or a cancer from a patient and I give them immunotherapy, are they going to respond or are they going to have adverse effects? And what we've done working with her is show that we can not only differentiate different types of cancer cells and also whether or not those cancer cells are responding to particular drugs, shown here in the colored image, but then within that tumor, you also have to look at your immune cells. And some of those immune cells include macrophages, that's part of your innate immune system. And your macrophages basically undergo different phase changes, right? There's different states. Similar to T cells. Your T cells, normally they're just kind of happy and scouring around, but once they see something that they want to fight off, they become activated. And figuring out those different phase changes of cells normally is extremely quite laborious and challenging, but we've shown with Raman spectroscopy, we can actually figure out just very quickly and even dynamically what those state changes are. So by looking at the different cancer cells and the different immune cells, which together form the tumor immune microenvironment and the cells that are part of the samples that are resected from patients, we've shown that we can now start to actually predict how patients are going to respond to different immunotherapies. So here's just the workflow. I will mention that here what we did is basically look at 33 patient drug outcomes, and these are actual patients in Stanford's clinic, and showed that our model could accurately, and without having ever seen these patients, predict 30 out of 33 outcomes, having trained on historic data. We were looking at prospective data, and then for the paper to come out, we actually had to wait and see how the patients fared. So I think this is pretty exciting for the future of actually treating patients with the right immunotherapy. So I'll wrap up and say I have some students who are out there at the posters. I encourage you go see what they're working on, but they're taking this platform now and thinking about how it can be deployed for environmental sensing, like on autonomous underwater vehicles, or thinking about things like wastewater-based epidemiology. And really what our vision is to enable these scalable solutions across personal and population health to really understand biology. And I'll leave you with this one calculation that is based on thermodynamics that says the information transmission rate of the biosphere is 9 orders of magnitude larger than that of the technosphere. So that means biology is communicating with other biological organisms at rates that exceed that of us communicating on our phones as a population by 9 orders of magnitude. So that is a ton of information with which to learn about the world and improve the world, and that we can't access now. And what we're doing is trying to develop basically the silicon nanophotonic hardware and the ML and AI algorithms that can really give us insights into this information communication. So I will leave you with a picture of my awesome lab. Many of the students are here today. I hope you get the chance to meet them. They have done all the hard work. Thank you so much for your attention, and I am happy to take any questions.
It's a great talk, Jennifer. Since Raman and IR are complementary, so are you thinking of combining with IR spectroscopy to get even more information?
Oh my gosh, amazing question. IR is great, and actually we do a lot of chemical transformations where we use FTIR spectroscopy to understand different intermediates. So you are spot on. For a lot of biological applications, kind of depending upon the setting, IR can be challenging because water absorbs so much in the IR. So Raman, if you design these sensors, they work quite well in a lot of the water transparency region, so you don't have to worry about IR absorption.
Hi, great talk. I was just wondering if the microbial identification assays were fast enough to be relevant to sepsis.
Ooh, I missed the first part. Which assay?
The microbial identification assays.
Oh, yeah. I would say sepsis is a great example. It's actually how we started the project, working very closely with someone in clinical microbiology. I would say, in principle, it could be. In practice, there are a few technical challenges, one of which is, in sepsis, conditions accelerate so quickly. So, every minute matters. And generally, you only have a few colony-forming units or a few bacteria per milliliter of blood. So I do think there's the possibility for folks who don't necessarily just want to look at the bug, but want to look at the host response as well, to potentially get information about how the host is responding very quickly. I think if you were to actually start thinking about having to digitize blood samples, and we have thought about that for other applications, it might be challenging to diagnose sepsis just based on looking at the bacteria.
Please join me in thanking Jen again. And real quickly, I want to introduce our next speaker. We were so lucky to recruit Professor Colin Ophis to our department 2 years ago. He was previously a staff scientist at Lawrence Berkeley National Lab, where he pioneered the development of several transmission electron microscopy protocols and methods. As recognition for his work, he's been awarded the Burton Medal from the Microscopy Society of America and the Early Career Award from the Department of Energy. And in addition to his research highlights, he also has this passion for scientific visualization. And he's founded and serves as the editor-in-chief of a new journal about ways to interactively communicate our science. That journal is called Elemental Microscopy. Please welcome Colin.
Okay, yeah, thank you so much, Sarah, for inviting me to give this talk here. And thank you, Jen, as you'll see in a moment too, for being an excellent collaborator and a hard act to follow, I have to say. So if you guys were expecting no AI in my talk, sorry, it's right here in the title. So we're going to be covering it too, because it really is in the air, as Jen says. So my group is focused on imaging materials down to the single atomic level here. And our main tool is transmission electron microscopy, or scanning transmission electron microscopy, STEM as we call it here. And so, for example, you can measure all these different information channels using this one single instrument. It's like a synchrotron in a microscope, right? So you can measure inelastically scattered electrons to do spectroscopy, similar to what Jen was saying. You can measure emitted X-rays to do a different type of spectroscopy. And I'm going to show you several of the direct imaging and diffraction methods that we use in my group here. So this is the flagship instrument that was obtained primarily by Jen, but many others here at Stanford, the Spectra, installed in our current Nano at Stanford lab here. And soon we're going to be getting another flagship instrument, this Neon. I don't have a picture of it because it hasn't been installed yet, but if you get a chance to go do a tour down in the Deep Lab, it's a very impressive facility that we're building here at Stanford to house these new instruments. And this is just a photo of me visiting the Canadian Center for Electron Microscopy, where they have a very similar instrument here. Okay, so let me give you a brief history of electron microscopy and atomic resolution imaging. So this is an image from my birth year, actually, showing silicon nitride down to essentially lattice resolution, about 1.6 angstroms. And after another decade and a half of research, we were able to push the resolution down to about the 1 angstrom level. So you can see it's a sharper image of this hexagonal lattice, but this is not the ground truth of the sample. It wasn't until the newest generation of microscopes, like the Spectra I showed you, that we were able to see the actual ground truth of this sample. And if I zoom in, you can see the importance of resolution. Even these small improvements, a factor 2, factor 1.6, you can now go from this hexagonal ring of blobs to separating those into the brighter silicon and the dimmer nitrogen here. So if I overlay the atomic structure, you can see the previous generation of microscopes was giving us lattice resolution, but now we're down to the single atom level here. So I want to quickly explain how we were able to achieve that image. The first technology that my group also works on is aberration correction. I'm wearing glasses. They can parabolically focus light down to a point, but unfortunately electron lenses have this intrinsic blurring. It's called spherical aberration, and it was responsible, for example, for those blurry images in the Hubble Space Telescope. We had to send a NASA mission up into orbit to install an aberration corrector, and believe it or not, the optics required to do aberration correction in TEM is even more complex. We've gone from 6 or 9 lenses in the microscope to hundreds of lenses. Another technology that I'm going to be talking a lot about in this talk is direct electron detectors. This is a biological image showing the difference between using a CCD to capture the electrons versus a CMOS chip that's open to the electron beam, where we're directly counting the charge induced by those electrons. And you can see it's a much, much sharper image. But there is a third technology that I would argue is equally, if not even more important going forward, computational imaging. So this is a movie of what the raw data looked like that generated the image in the upper left. So this is what we would call a focal series, where we're changing the defocus of the microscope and we're collecting the sort of different ray paths under different imaging conditions, and then using computer algorithms to synthesize it together into an image. So that previous 2 images I showed you were essentially photoplates and a CCD, but now it's a computer that's taking in the raw data and reconstructing the image here. So the fourth thing I have to mention, because I am an electron microscopist, sample preparation, equally important. If it's not thin enough, it's not electron transparent. And so that is something that Stanford has also invested heavily in, and I am very grateful for that. Okay, so now I want to move to the main technique of my talk, scanning transmission electron microscopy. So conventional STEM, we would raster a probe across the surface, and you can see this big donut-shaped detector which would collect a single value at every probe position. We move the probe, record another image, move the probe, repeat. But if you look at the bottom, the diffraction pattern is much, much more complex. It has a huge amount of information. And now we can use those direct electron detectors to capture that full image of the diffracted probe, move on to the next probe position, repeat, repeat. So the resulting data sets are enormous. They're 4-dimensional. We have 2 spatial dimensions and then 2 reciprocal space dimensions here. And that gives the technique its name, 4D STEM, for the 4-dimensional data. So this is one of my earliest 4D STEM experiments. This is showing tungsten disulfide in our team microscope at Berkeley. And this is giving sort of a picture of what the diffraction patterns look like. But here I've averaged 40 by 40 probe positions. If I take a step back and show an average of 7 by 7 probe positions, you start to grasp the scale of the data here. This experiment ended up being about 420 gigabytes, and this is what we were able to do in 2018. Now we can record a terabyte of diffraction data every 6 seconds. So the data rates are completely out of control, and this is essentially the main challenge of the modern data-rich electron microscopy technique families. It's how do we turn all of this data into actionable material science information. Okay, so just to show you an example of this, if we had a crystalline sample and we were to scan the probe across the surface, you can imagine that these diffraction patterns are fingerprints of the local phase and orientation. And it doesn't just work for crystalline materials. If I take a toy model and I randomly move all the atoms in this sort of linear path and scan the beam through, what you'll see is the two-dimensional grid of peaks, Bragg peaks as we call them, is replaced by these characteristic amorphous halos. So you can imagine that we can use computer algorithms to identify these different structures and invert or infer what the sample looked like here. So let me show you a real experimental data set. This is gadolinium titanate. It's a material that was being studied by Los Alamos for fusion reactor cladding materials, actually. And so what they did to study the resistance against ion irradiation is they put it in the reactor, and what this causes is amorphization of the material. Deep, deep, deep down into the sample, you're randomly displacing all the atoms and removing that crystalline order structure. Now, of course, they want to have a lifetime of these materials of, say, 20 years. We don't have enough time to wait, so instead they anneal it at higher temperature to see what the sort of long-term reaction of this material would be. And what you see is you recrystallize into the amorphous front, but you also nucleate a bunch of randomly oriented polycrystalline grains. So what I want to show you is all the diffraction patterns that we record from a sample like this. And just by using what you saw in the toy models in the previous slide, you can see we're able to figure out what regions are single crystal pyrochloride, the original structure, recrystallized fluorite, or these amorphous or disordered regions of the sample here. So this is an example just showing a whole bunch of different information we can get from a single experiment, one data set, but many, many different channels. The phase, the crystalline orientation, the degree of order and disorder, and many other techniques here. Okay, so for the main 3 studies that I want to show today, I'm going to zoom in down to atomic or near atomic level here. So if we were to do that same 40-stem experiment here, but on a magnified scale, a much larger convergence semi-angle, this is a simulation showing what it would look like for tungsten disulfide here. And you can see the crystalline hexagonal lattice in what we call the ronchiogram, the far field diffraction pattern of the probe. So what I'm going to show you is how we can use adjacent probes, the overlapping information, to computationally invert the sample structure. So we start with a guess of the probe, then we have an empty object because we don't know what it is yet, and then I put a red dot everywhere we have one measurement, and we forward simulate the physics to see what the scattering would look like, and we compare it to our measured diffraction pattern. And of course they don't agree because we haven't solved for the probe or the object yet. But if we do something called backpropagation, which is what we use in machine learning to train our networks, we can measure the difference and update our guess of the probe and our guess of the object. So if we repeat that 2 iterations, 3, 4, 5, we will very quickly converge on a very accurate guess of our probe and a very accurate guess of our object here. And this deconvolution is why ptychographic 40-stem is such a powerful imaging technique. So let me give you a quick case study. One of the last things I did at Berkeley before I left. This is an Alex Zettel sample. It's a double-wall carbon nanotube with a zirconium tellurium nanowire encapsulated inside. And their lab had formed many of these different structures using their chemistry techniques, but nobody knew the structure of any of the more complex nanowire nanotubes here. So of course, if I show you that technique map here, ADF is that single pixel channel I showed you before. This is unfortunately a high atomic number sensitive technique. So we can see the zirconium and tellurium, but we can't see the 2 outer walls of the carbon nanotube, right? If we switch to a 4 pixel measurement, DPC here, we are able to get the carbon, but we're blurring out the sample. It's only when we do the full computational pipeline here, this ptychographic method in the lower right, that you can see a very sharp image of all the individual atoms in the nanowire and in the nanotube here. So in order to solve the 3D structure, though, we have to do what I showed you before, solve the probe and solve the object, but over many different tilt projections of our sample. And so this is all of the different solved quantum mechanical electron probes. And these are the different quantum mechanical scattered waves of the nanotube nanowire at all the different tilt angles. So you can start to see the complexity of even a tiny structure like this. And this is, again, a very unique structure, right? If they grow a nanotube or nanowire with slightly different diameter, they would get a different crystal structure. But we're able to use computational methods similar to an MRI or a CAT scan to turn those two-dimensional images into a three-dimensional map here. And you can see the resolution that we're able to achieve. Single atom level in 3D here. And of course, in material science, if you knew where every single atom was, you could answer almost any question that you would want to know about the sample. So again, just to reiterate here, this is what the nanotube looked like in 2D, and I tried in vain for years to solve the structure, to guess what the 3D structure would be from 2D images like this. And it wasn't until we solved our first one that I understood why it had been so difficult here. The structure is immensely complicated. Not only do you have this double-wall carbon nanotube, it isn't even circular. It's stretched into this ellipse, right? I always imagined when we saw the two-dimensional projection that it was a circle in projection. It's not. So that was the first problem. The second problem is inside the nanotube, it wasn't just one crystal structure or two crystal structures. There's this distinct core and shell region, and then individual chains of tellurium atoms actually following at the top and bottom. And I would never have been able to infer what this 3D structure was from a single two-dimensional projection here. Okay, so I'm going to show two quick studies here, but now this is what we're doing now. Thanks to the AI revolution, as Jen showed in her talk. There are many techniques available to us now that we couldn't use previously, and chief among them that I'm going to show you is the idea of using deep priors here in our loop. So what we're essentially doing is we're using a generative AI technique to create our object, and then we're using forward physics models to simulate what our measurements would look like. So that's our predicted experimental intensities. The loss function is the difference between our measurements and our predictions, as Jen showed. So then again, we use backpropagation to update not just the weights of our machine learning network, but also the parameters. For example, in that 3D experiment, what the shifts are between our measurements, the tilts of our samples. And this is a very powerful technique for virtually any inverse problem, because you can see this loop, the only thing that's going to be different in different sciences is that little box called physics model. So as long as you can write down the forward problem, this is a framework that you can use to solve these problems. So this is an example using data on a metal organic framework, and this material is so beam sensitive that you have to be in the tens or hundreds of electrons per angstrom squared. And the upper right is showing what one of those measurements looks like, right? It's very sparse. We only have a couple hundred or a couple thousand electrons in each pattern. And if you do a reconstruction, that's what we're showing here, this pixelated object, it's very noisy and very blurry. But by using this deep generative prior approach, we can get a much sharper view of our object. And we actually use it for both the probe and the object. If you use it for just the probe or just the object, you get a much blurrier result here. And so I just want to zoom in and look at each one of the structural units here and loop through them. You can actually see the two bright dots of the two zirconium atoms in projection next to the two dim dots of single zirconium atoms. And in between, it's empty space. These metal organic frameworks are mostly vacuum. And this is actually one of the reasons they're so beam sensitive here. I think I can play this again. What I really want to emphasize too is on the chains, you can see those blobs. These are essentially the benzene rings, right? The most beam sensitive parts of the chain. And we're now able to get down to the sort of single atom level and make measurements here. So the other thing I want to show is a quick slide on biology, again, because I'm following Jen. This is a Phi92 bacteriophage. This was a collaboration with Henning Stolberg's lab at EPFL. And we're using my group's techniques and software and applying them directly to biological data sets here. And so this structure is just essentially it's a virus that attacks bacteria. And I don't want to go into the biological details because I don't know them, for one here. But I just want to show that the pixelated reconstruction, right, you can immediately see the image quality of the deep generative prior versus the pixelated one. And it's quite robust. As long as you use one of these physics-informed machine learning models, you're able to get a much more efficient and often much faster reconstruction of your sample. Okay, so let me show you one last experimental example, again returning to 3 dimensions here. So this is something called an implicit neural representation, and this is, for my money, just one of the craziest ideas in machine learning. Instead of representing our image as a grid of pixels or our volume as a 3-dimensional grid of voxels, what if you just do away with that? You make a black box network here, and you just have 3 inputs. What's the x, what's the y, and what's the z coordinate here? And then you ask it, what's the intensity of your object? So if you want to apply this to 3D tomography, you can imagine every pixel in our measurement is a ray that's tracing through our 3D volume. So we simply ask the network, what is the value here, 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 sum it up in a projected vector, and that's what we compare against our experimental measurement. And then we use that to update our object weights here for the generator. And of course, we also update the pose of the object, the rotation, the tilts, and this makes a huge difference, as you're going to see. So this is, again, a GenDion sample. This is done by my student Cedric and our shared postdoc Saren. And this is a 3D reconstruction of one of their catalytic nanoparticle systems sitting on a support, a titania support here. And so on the left-hand side is the INR reconstruction. The right-hand side is the conventional one. And you can immediately see in the conventional reconstruction massive artifacts, right? The particles have these little wings that are sort of taking off. And that's because when you zoom down to the single atomic level, tilting has to be extremely precise. Our stages are mechanical, right? So you're going to have a 1 degree error here, a 2 degree error there, and this micro misalignment leads to these artifacts in the reconstruction. But it goes further. If we take this volume and we slice it in different directions here and then zoom in on one of the slices, again, on the left-hand side is the machine learning-driven reconstruction. Right is a more conventional algorithm here. And I just want to highlight the massive difference between the two. When you're studying something like catalysis, it's actually the triple points that are often the most important structural components, where the gas phase can meet the catalytic nanoparticle and the support. Because catalysis is a multi-step process. You have to have the gas molecules able to land, attach, disassociate, or go through some chemical reaction, and then often migrate over the sample surface, recombine or change or desorb into the gas phase. And if you imagine the conventional reconstruction is all you have to go on here, you can see the nanoparticles, but the support constraint is essentially completely blurred out by these artifacts, by these extra errors, and that's just caused by missing information. We can't fully tilt the sample 360 degrees, and our measurements are not sampled at 0.1 degree increments. We have a limited number that we can perform here. So the last thing I want to mention in my talk here is I am an absolute proponent of open source science here. Everything that my group is developing here at Stanford, we are releasing open source data sets, open source tutorials, open source codes under our Quanti-M banner here. And thank you, Sarah, for mentioning my journal, Elemental Microscopy, which is also focused on open science communication here. And we're doing this under very friendly licensing terms because we want to help researchers, but also industry as well. And so now we're starting to work with industrial partners and get a lot of our methods and a lot of our software directly embedded into their pipelines, into their characterization, their metrology, et cetera. And so with that, I definitely want to thank all the different funding sources here and also comment too, I am an immigrant to America and I very much have benefited from government funding, right? So I really hope in this country that we can continue to support science with the, I would say, the best in the world support system and funding system and network of universities, right? America is still where all the best science happens and it attracts, as Sarah mentioned, all the best people from around the world, especially here at Stanford. And then, of course, I want to thank the students, especially Arthur, who's here presenting a poster on the deep generative prior work. The INRs, Cedric Lim is presenting a poster. And the previous work that I did at Berkeley was done by Philip Pels primarily, and he's now a professor in Germany. And so with that, I would be happy to take any questions.
Thank you very much for a very interesting talk. I'll ask a question more out of ignorance, but I was very pleased to see somebody talk about inverse problems, in fact, more than one person today. As speaking from the mathematical point of view, have you ever run into a problem where you either encounter non-uniqueness or no solution at all?
I've run into both of those. So the question was about non-uniqueness and not being able to find a solution. And I would say this is actually one of the dangers of a lot of these. Essentially, you can think of the machine learning, one of the things it does as non-local denoising. So it's using information from this part of the image to update what it might look like over here, right? And so if you do that using conventional computer vision techniques like principal component analysis or some of the early machine learning work, you have to worry about hallucinations in your data, right? And a lot of that is caused by non-uniqueness. And in 3 dimensions for tomography, it's a particularly difficult problem Because especially if you just imagine a two-dimensional projection, it's extremely non-unique, right? And even in under 3D conditions, you need a lot of tilts to be able to identify the specific object. So this is actually one of the things we're working on now. How much information do you need before your solution is unique? So do you need 5 different tilt projections, or 11 projections, or 100 projections? And unfortunately, the answer is, of course, it's sample dependent. Very, very complex samples that have a lot of heterogeneity, a lot of small length scales with a lot of variety, you just need to collect a lot more information. But luckily, statistics in particular, through techniques like cross-validation, which have also been applied in machine learning, give us the tools to know when we can be relatively certain that the thing that we're measuring is unique. The other question about not being able to find a solution, that's a massive problem for these ultra-low dose experiments. So if you were to measure something with 0 electrons, of course you can't solve it. If you were to measure it with 100 electrons or 100,000 electrons, there is some point where these complex inverse problems become solvable. And again, this is a question that we have to use statistics to identify. So in inverse problems, I would say especially non-uniqueness, but also solutions not existing or blowing up, it is a huge problem. And we do a lot of cross-validation, a lot of forward simulation, like complete known ground truth in order to validate these methods. Nobody is more skeptical about machine learning than me. I had to be convinced that this was a good way forward.
Please join me in thanking Colin again.
Well, one of the elements of the Nix Lecture event that's most interesting and most pleasurable for us is to welcome back one of our distinguished alumni to present and describe events from their career and to really see the arc of the career of someone who at an important point in the early part of their journey were here at Stanford in material science and engineering and have gone on to do very impressive things after that. And certainly that's the case for our distinguished alumni speaker here today, Dr. Stephen Streiffer. So I have the great pleasure to introduce Stephen for this presentation. At the current moment, Stephen is the director of the Oak Ridge National Laboratory, one of the premier organizations for scientific and engineering research in the world, with 7,000 employees and many exciting research programs, including neutron scattering, advanced scientific computing, nuclear power research, isotope manufacturing, and of course a lot of material science. So he's sort of in his element in his job at Oak Ridge, and I'm really looking forward to his talk here today. But before that begins, I'll say a little bit about Stephen's background. He earned his bachelor's degree in material science and engineering at Rice University before coming here as a PhD student in the late 1980s, and he earned his PhD in 1993. Actually, that's when I became aware of Stephen. Years before we ever met, I was actually working on something quite similar in my own PhD thesis on the other side of the country, right around the same time, reading Stephen's papers and being incredibly impressed by his research. And it was very inspirational what ultimately I did for my doctorate and some of my subsequent work. And I had the great good fortune to work with Stephen in various ways in the years since then. So that was before I even knew him. He spent some time as a research professor at North Carolina State University and then joined Argonne National Laboratory in 1998 as a materials researcher. And there he was engaged in a really fantastic set of studies of really fundamental in their nature of the structure property relations and synthesis of ferroelectric thin films, and really trying to understand the connection between all of the different physical phenomena associated with ferroelectricity and the domain structure, orientation, strain, many important parameters. And this work was very influential, once again, and led to his election as a fellow of the American Physical Society in 2007. At that point, Stephen made a change in direction and began moving into increasingly important leadership roles at Argonne. In 2015, he became the director of the Advanced Photon Source, which is a tremendously important and successful scientific user facility in the United States and really worldwide. And he was also associate lab director there for photon science. And then in 2020 became, in addition to his role at APS, he became a deputy director of science and technology at Argonne Lab and held that position for 2 years. And then came back here to the farm in 2023 as Stanford's vice president for the SLAC National Accelerator Laboratory. That's a really important role. As a contractor, the university holds the contract with DOE for operating a major national laboratory at SLAC, and the Stanford VP's role is to basically be the representative of the university and effectively holding the lab's feet to the fire to make sure we do as we should in honoring our contract, and also, of course, supporting the activities of the lab and the connection with DOE. And I think Stephen did a fantastic job in that role, and then the next year became interim director of SLAC. He held that position until late 2023, at which time he became director at Oak Ridge. At Oak Ridge, he's engaged in a wide variety of pursuits in line with the mission of that laboratory. And one aspect of that is that he has been recognized just in the past year by the American Nuclear Society with its presidential citation for advancing nuclear energy and innovation. So with that, I give you Stephen Streiffer.
Thank you, Paul. I really appreciate it. So it's just an absolute honor to be here with so many friends and colleagues. It's always great to come back to the farm and see what's going on. It's also just really humbling to be on the program with the distinguished list of speakers today, and I'm really honored for the invitation to come and talk. I will warn you, I have several anecdotes that I'll share with members of the audience. Those anecdotes I'm sure are true, but I am not sure they're accurate. So wait on me in terms of exactly what I say or don't say. Let me just jump in, and actually as a testament and as really the sincerest form of flattery for Stanford. What I chose to talk about today was just going back to 1993, the period in which I got my PhD, and use a comparison of that to where we are in a number of areas in science and technology, just to try and get a sense of how things change. And as a matter of fact, those of you who are associated with the high TC superconductivity era that was occurring as I was a graduate student, this title itself is actually cribbed from a talk that Mac Beasley gave, as he was trying to get the graduate students really oriented to what science was like, rather than what we thought it was like during this craze that was high TC. And that was very important. Paul has already gone over a lot of this, but it'll set some of the context for the talk. As Paul has pointed out, I got my bachelor's degree at Rice in 1987. That turned out to be very significant with respect to the faculty that was at Rice at that point in time. Then had the fortune to do a von Humboldt Fellowship at the Max Planck Institute for Metallforschung in Stuttgart, working for Manfred Rühle. Turned out one of the other directors at that point in time was Ed Arzt, and that turned out to be an important connection that I think has actually played out through this community as well, particularly for Bill. Moved from there to North Carolina State for a few years. That was actually where I first in person met Paul. So we were both working on, at that time, an ARPA project. They had dropped the D for a short period of time, trying to integrate barium strontium titanate into semiconductor memories as a dielectric. And my takeaway from that, which I think, again, will really appeal to everyone in this audience who is often very focused on the translation of technology into real commercialization. We met with the vice president for technology at Micron about that project at one point in time. And it became very clear that the academics at that stage were working on what to us looked like integrated semiconductor devices, but we're not even in the same universe as an actual integrated dynamic random access memory. And at that point in time, we really should have just stopped the project. There was no hope it was ever going to succeed. So I think that really speaks to the connection that Stanford has been very good at bringing both the academic world and the private sector world together in a way that what we do is actually meaningful. It's very easy to think that you're working on something important, but somebody who actually knows what it takes to get something in the marketplace can often very quickly look at it and say, not a chance in hell. And that was a very important lesson for me. Then moved to Argonne National Laboratory for just about 24 years, and that will be an important thread through this story. And there are actually more ties back to Argonne from the Department of Material Science and Engineering here than I think many people realize. Paul was very gracious in his remarks about me. I'll quote someone else again that you all recognize with the saying that if I see further, it's because I stand on the back of giants. The collaborations that I've had the benefit of having through my career, including a number of people that are sitting over on this side of the room, have really been a team effort to, I think, achieve some very significant things in both our understanding of oxide films, as Paul has already pointed out, but also push the forefronts in some of the techniques and the facilities that we all depend on to really advance our science. It was a great year and a half here. It was great to be back, but unfortunately I couldn't resist the lure of going back to Oak Ridge National Laboratory. It's really a very large laboratory, which is quite exciting. I'll also point out that there are individuals who've been connected through the Department of Material Science and Engineering and through Stanford University in various ways prior to me, and just wanted to call them out as well. So I'll start on the right here. Jeff Wadsworth was a postdoc for 4 years, I believe, with Oleg Sherby, working on various mechanical properties of materials. He became the director of Oak Ridge in 2003 through 2007 before he actually moved up to Battelle Memorial Institute as part of the corporate management there. And he succeeded Bill Madia, who was the first laboratory director underneath the UT Battelle management umbrella, a co-operation of the laboratory by University of Tennessee and Battelle Memorial. And of course, Bill came to Stanford when I believe Persis Drell was the director, as Paul has pointed out already, to establish this role of vice president for SLAC National Laboratory as a way to make sure that the university was actually overseeing the lab, and as Paul points out, holding the lab's feet to its fire and making sure that it was delivering on the Department of Energy mission. Bill has had an enormous influence on a number of us that have come up through the system. Bill also had an enormous influence on the SLAC campus itself. Bill was an aggressive fundraiser, which is actually very interesting in the National Laboratory arena. And his relationship with the Arriaga family led to a number of new facilities on the SLAC campus, mirroring some of what we're in today. So that connection is actually quite amazing. I apologize, I'm contractually obligated to show a set of slides now as the director of Oak Ridge. I'll try and get through these reasonably quickly. But Oak Ridge National Laboratory started out as part of the Manhattan Project, actually in 1943 as the Clinton Engineering Works. And the element of what is the larger DOE presence in the Oak Ridge Reservation is centered around what is known as the X10 location, or the graphite reactor, which is the second reactor after CP1 in the University of Chicago to go critical as part of the Manhattan Project. And what was fascinating about that, the goal of this reactor was to actually demonstrate that you could have a reactor that would go critical, that the fission in that reactor associated with uranium would produce plutonium that could then be separated and used as part of the Manhattan Project. One of the amazing things about this to me, this was actually, first off, it was a public-private partnership, words we hear a lot today, but this was actually a collaboration between University of Chicago, who provided managers and scientists, and DuPont, who did the construction. And this was a design build. They were in there, they were building this thing, they were finishing out the design for something that had really never been done before, that really was part of the magic that made this thing happen. Also amazingly, from when the first shovels went in the ground to when this reactor went critical was less than 9 months. Can you imagine building a nuclear reactor in 9 months today? Now, the flip side of that, which I always like to remind people, is more than 80 years later, we're still cleaning it up. So I hope that there's a better balance between building a reactor in 9 months and cleaning it up for 80 years, and building a reactor in 80 years that we don't have to clean up after. And I think that's actually part of the mission that we have as an international community right now. Quoting Ernie Moniz, the energy solutions that we need as a planet really are all of the above. I do believe that fission and fusion are part of that. I'll say a few words about that in a minute. But this was really part of the birthplace for that. The other thing I'll say, which I always get a chuckle out of as being a member of the National Laboratory Complex, Oak Ridge was founded in 1943 as the Clinton Engineering Works. Argonne National Laboratory was founded in 1946, coming out of the Met Lab. And then, of course, we had the effort at Lawrence Berkeley National Laboratory coming out of the Calutron effort and Lawrence's work there at the university. There's an interesting set of advertising criteria that each of these labs, advertising slogans that these laboratories use. Argonne claims it is the oldest national lab, if I get that correct. Berkeley claims to be the first national laboratory, and how those are not the same thing actually kind of boggles my mind. And Oak Ridge was actually formed in the middle of those two. So just an example that everybody will find something to hang their hat off of. The laboratory really has focused in what I would like to say are 4 different areas, really underpinned by our role in the national security portfolio as well. The laboratory has been a leader in high-performance computing for decades now. One of the stories I was telling last night was there was this small gaming chip manufacturer that came to the laboratory in the mid-2010s, basically trying to understand if their chips could be used for scientific computing. So Oak Ridge National Laboratory worked with them basically to change the instruction set on those chips, then was able to build a machine that incorporated those chips with some other vendors as well. And that really led to the birth of what we know today that's used by the hyperscalers and the other tech industry around the world for running AI, which we've heard a lot about. That was the birth of using GPUs in high-performance computing, and that company was NVIDIA. So NVIDIA's success today really has its origin in the partnership with the Department of Energy laboratories and trying to keep the US at the forefront in high-performance computing. We continue that trend, and I would say right now it's really focused on what I like to say, getting off of the GPU bandwagon or the treadmill. We really need to find new methods for computing that are much more energy efficient, that can open up the ability to calculate problems in ways that we can't do with traditional von Neumann computing, things like quantum and other mechanisms like that, and leapfrog where industry right now is so that we continue this trend of really being able to deliver on the science and technology promise of computing. I've already mentioned the nuclear mission of the lab. We're still very deeply involved in fission, trying to get the nuclear renaissance really relaunched not only in the US but around the world. And then extending out a little bit further, looking at fusion as hopefully a viable energy source sometime around the mid-2030s if we can stay on the technology path. And then associated with that, the laboratory actually has a very strong isotope program. I do apologize to everybody in the room. If you've ever been through TSA and have been swiped for explosives, that was us. We actually make the isotope that goes into those machines that are used as essentially the energy for the spectroscopy to look for the compounds that are common in explosives. We have, as Paul has already mentioned, a very strong material science program that's really based around our applied energy and our nuclear work. We operate something called the Manufacturing Demonstration Facility, which works very closely with industry in teaching them how to solve problems that they may have in applying advanced technologies to particular product lines. And there's a huge economic benefit that goes out indirectly to the nation as a result of that. And I was doing a little bit of the back of the envelope calculation that work that's gone through the MDF over about the last 10 years or so turns out to have touched something like 1% of the gross domestic product of the United States. That's a pretty amazing figure. That's only part of the National Laboratory Complex. And then finally, moving it up to the systems level, one of the things that we have a very strong focus on is looking at things like cyber-physical security of the grid. Taking the integrated energy systems that are becoming increasingly complex, becoming increasingly interconnected, and understanding how we can operate those more efficiently, how we can operate those in a more secure way. And then looking at even things like building technologies in a way that we can make buildings more energy efficient, more livable, and more constructible. And then leveraging off of all of that, given our history from the Manhattan Project, just as an example, if you're good at nuclear fuels, you're good at nonproliferation. So there are lots of ways that we leverage these strengths into the national security portfolio for the nation. Just a few facts and figures. I won't dwell too much on this, but this gives you a sense of the scope of the laboratory. One of the things for this particular audience is I'll call your attention to the right hand of the figure where we talk about the number of patents that we have each year. And of course, for this audience, that will seem like an incredibly small number given the size of the laboratory. And again, that's because the way that we do commercialization, the way that we impact the economy of the US is very different than you see in academia. I think it's one of the lessons that we have to learn how to describe better because your taxes pay for the operation of the laboratory system. And the impact that the labs have on our everyday lives and on the long-term security and economy of the nation is really profound. And getting that message out in a way that people understand that better is something that all of the laboratories are really now working on. Just a couple of examples of some of the things we've been doing recently, ranging from really fundamental nuclear physics, looking at neutrinoless double beta decay, which is ultimately aimed at trying to understand the asymmetry between matter and antimatter in the universe, to things that are much more practical. One of my favorite examples is here down on the right, where we have worked with some of the nuclear industry that is in the Oak Ridge area to develop new ways that we can actually make forms for concrete in a way that is much more rapid, much more cost effective, and can be deployed very quickly for very complex structures as well. So the example there that's shown is one from a company called Kairos Energy. They're building a molten salt reactor that, again, is derived from work that actually was done at Oak Ridge in the 50s and 60s. The problem with this is they need to build very complex concrete forms that effectively make up the bioshield around the reactor, basically a radiation absorption medium, not a structural medium for the reactor. And to do this, they were trying to come up with ways where they could actually essentially make a hexagon that would surround the reactor vessel and do that in a way in which the concrete was interlocking so it didn't have to be grouted. And these are very tall. Ultimately, these are going to consist of two 20-foot sections that are stacked on top of each other. You can't do that with plywood. If they were going to do this with steel forms or metal forms, it would have taken about 6 months to design them and probably about the same order of magnitude of time to actually get those forms built. We had forms from when they came to us drying on their foundation, with concrete in them, drying on their foundation in 14 days. So this is the way in which the people in this room can really accelerate innovation and do things that will move the country forward at a rate that really is just unprecedented. And of course, we've heard a lot about AI already and how that will leverage what we're doing. I don't have to explain to the audience the importance of what that is. That's something that the laboratory has been deeply involved in for quite some time. It used to be that in the national laboratories was a strong focus on a discipline called automated reasoning. We now know that as machine learning and AI. One of the things I like to joke is the national labs did AI before AI was cool. We really have been working on that for about 30 years. And as a matter of fact, the first neural network I used as a researcher was actually in 1993. So some of these technologies have been around for a while. It's actually really interesting to see them go ballistic as we move forward. Just a couple of words about the nuclear renaissance. It's easy to forget that very similar to politics, all energy is local. And it turns out if you look at the East Tennessee region, wind and solar don't work because of the location and the geography. And you don't want to ship energy around. So the energy solution in East Tennessee in particular is probably nuclear if we want to get to a carbon-free solution. And as a result of that, there are a host of startups in the area that actually have reactors coming out of the ground. So these are not paper designs anymore. These are things that are actually getting built as demonstration units, which are moving forward very quickly in a way that's just amazing to see. There's about $12 billion worth of investment in the Oak Ridge area in the last couple of years, creating about 3,000 jobs. And it's not just the reactor vendors, it's also the fuel suppliers. So one of the tipping points we're seeing is the individuals, the entities that were interested in building reactors were looking at the fuel suppliers and saying, I don't know if we should build a reactor. I'm not sure if we're going to have the fuel. And the fuel suppliers have been looking at the reactor folks saying, I'm not sure if we should build fuel because we're not sure if you're going to build the reactors. We seem to have broken that deadlock. And there are companies like Orano, which is a French company, that's investing $2 billion into the region to start producing high-assay, low-enriched uranium fuel for its advanced reactors that will really start to power the fleet in a way that the U.S. hasn't done in decades. Just a quick snapshot of some of the things that have happened since I got my PhD in 1993 that set some context for that period of time. I won't read these all, but there are a couple things I think are very interesting here. So I mentioned the Woodstock of physics, the high TC craze that I as a graduate student came into. Very interestingly, I got my comms people helping on this talk, so they went and found a picture of the Woodstock of physics. And it's kind of small, but the guy on the left there is actually Mac. So it's interesting to see how this thread has run through the community in a way that's still sustained. Now, the object lesson that I have for you there is I came into graduate school thinking it was always like that. And graduate students hauling mattresses into the attics of academic buildings, working for 24-hour shifts to try and get ahead of everybody. That was not the way that science used to be done. That was also the time at which the World Wide Web was created. We talked about this a little bit earlier as well. And internet communications were just starting to become feasible. And that has really transformed the way that we think about science. The pace that we moved during the high TC revolution, where we got used to things like preprints, communicating conference proceedings and results before the publications came out, and the ability to transmit information that didn't rely on people actually being in the same room. During this period, that was really where that transformation happened. There's another thing I'll point out, which gets back to Colin's talk earlier. The Hubble telescope was launched in 1990. This will get a little bit ahead of myself, but the astronaut who was the one who put the corrective optics onto the Hubble telescope in 1993 was a guy named Jeff Hoffman. Challenger exploded in 1986. The astronauts down at the Space Center in Houston, just south of Houston, all of a sudden found themselves having to find things to do. Jeff Hoffman decided he would get his master's degree in material science at Rice as I was finishing my work there. So I actually had the great displeasure of somebody who was a wet behind the ears undergraduate of working with a seasoned NASA astronaut. The program at Rice was relatively small. There were a lot of classes that were both graduate and undergraduate classes that were taught at the same time. Being in a room with Jeff Hoffman, pretty intimidating for an undergraduate. The guy was damn smart, and trying to keep up with him was pretty incredible. But this just gives you some of the thread of what was happening during that time. Some good, some bad. A couple of things that really impacted the university during that period of time. Of course, the Loma Prieta earthquake happened. We had the fires over in the Berkeley Hills in 1991 that really reshaped the landscape in the East Bay. So it was actually very much a transformation, not only for the department, but for a lot of the ways in which we do science and engineering, and in many ways for the university as well, that set the stage for where we are today. One of the things that I did to prepare for this was actually pull all the dissertations from a 10-year time period from 1987 to 1997 and try and take a look at what was going on at that time. Now, that's kind of a weird thing. You might ask, why 87 to 97? That's kind of a weird set of dates. That covers when I showed up and the people that I knew then to when the last people that I knew at the university as a graduate student were really finishing up their work and publishing dissertations. And Sean, it does turn out that Annalee was one of those people that was about 1997 or so. So we did actually catch, I think, many of the relevant people. But there's the traditional word cloud, which kind of gives you a flavor of what was going on. These analyses are always a little bit subjective. If you look at the silicon work that was going on, that really shifted over that time period from work that was on silicon bulk crystals and silicon thin films and integration with those to metallization with aluminum and copper, which of course might be mechanical properties, might be silicon technology. Those are the sorts of choices you have to make. The compound semiconductor work that was going on at that time was kind of split between gallium arsenide on one hand, looking at that as a semiconductor device, but a lot of work that was done in a couple of faculty members' groups on solar technology. So a lot of that was actually compound semiconductors for solar. One of the interesting things looking back on that is you can tell when Bruce Clemens was hired, because all of a sudden you had a spike in magnetic materials PhDs. That was about halfway through my tenure at Stanford. There were several other people that were hired after that that really got the magnetics program started at the university. And then a smattering of other things that you can see here. I was one of the 3% that was working on high TC. That included people like Daryl Shlom, Bruce Larson, Chun-Bum Ohm, and others who many of you will know. There were a couple of other things about this. There had already been a shift in the department, the work that was being done. About 2/3 of the dissertations from this time period were on thin films or surfaces, where the remaining were on bulk. So the department was already moving towards that as really a central focus. You'll notice a few things that are absent from that. Nothing on quantum dots. The first research on quantum dots in the department was about first dissertation was after 1993. Nothing on things like fullerenes. You can actually tell when Stig Hagstrom joined the department because that's where diamond became a part of the portfolio of research that then continued for a number of years. There's no biology here. And that's very different than we are today. So it's actually interesting to see how research has evolved over time, and the department has kept up with that. That's really amazing to see as the talks we've heard today. The other thing that was really interesting in looking at the dissertations is only 10% of the dissertation titles explicitly mention theory or modeling. And of course, that's very different than the world that we live in today. And as a matter of fact, during my time in the department was really the first point where the department started to get Spark workstations that were being used as part of the research portfolio that then set the trend for scientific analysis and scientific software packages that we see today. And one of the things I very clearly remember, Jim Bain, who worked for Bruce Clemens, was one of the first people to get a software package called Igor running on some of those workstations. He was working on doing some fitting of data that he got from SSRL. The time to do one iteration in the refinement was about 20 minutes. And if you sit down with your Mac today, you can still run Igor. It can run through a very complex model before you can blink your eye and actually do a convergence on just a standard optimization for that. So we really have changed the way that we work relative to where we were then in ways that it's very easy to take for granted. Let me now turn down to Bill. I can't be up here without telling a couple of stories about Bill. So first off, as I pointed out, I was actually got my undergraduate degree at Rice University, and I think the person who really got me into material science and engineering was this guy named George Farr. George was an early career faculty member who was at the university at that point. I had several classes with him, and it was really him who drew me into the discipline and also got me appointed at Stanford. And of course, George was a graduate student under Bill. If I recall correctly, George got his degree in 1979. Very interestingly, he then moved to Rice. After Rice, he moved to University of Tennessee and Oak Ridge National Laboratory for a number of years, and then in 2017 actually moved to the University of Tennessee. And one of the things that was really interesting about that was George invited me to give a colloquium at UT, the other UT, last year. It was really interesting to compare history. I'll get to a little bit of that in a second. But one of the things that was actually kind of embarrassing is George still had his grade sheets from when I was an undergraduate there. And getting back to Jeff Hoffman topped the grade sheets. I'll just leave it at that. I won't say where I was, but it was really interesting to see that comparison. And George, of course, and Bill as well, have been very connected to a number of people that I worked with in other ways. I mentioned my time in Stuttgart. People I worked with there, or at least interacted with, included Ed Arzt, Oliver Kraft, Chef Baker, Tina Shoy, Gerhard Dame, and others who eventually came back to the US, often in electron microscopy, often in mechanical properties, that are part of Bill's orbit as well. Now, a couple stories about Bill. Again, I promised you I was going to roast you. So there was a department event, and as the department I'm sure still has, one of the challenges we faced at that point in time was space. And illustrating Bill's sometimes sharp sense of humor, Bill got up in front of the department and said, as part of the faculty management of the department, he had been appointed the space czar. And then he got that glimmer that only Bill can have, and you could see the twinkle in his eye, and he paused for a second and said, and that means everybody in my group is now known as a space cadet. And that just is a sense of both the seriousness that he took, actually diving in and helping the department solve a very difficult challenge, but the light spirit of wit that he took to that I think was something that set sort of the culture of the department. Second story which shows Bill's modesty. Those of you who are material scientists will recognize the term the Thomson tetrahedra. One of the things that distinguishes material scientists from applied physics and physics is we don't usually name things. People in physics tend to name things after the discoverers. It's actually relatively rare in material science. But one of the things that Bill sort of realized during the time frame that we're talking about here was the Thomson tetrahedra was correct in defining where the slip planes were in the FCC lattice, but it was wrong about the connectivity. And the dislocations actually moved on what many of us started to refer to as the Nix pyramid, which were the geometric shapes that were on the other side of the Thomson tetrahedra. Bill did not like us referring to that as the Nix pyramid. Just again, getting back to the point that it was really a seminal observation that described a lot of the mechanical properties and was a much better way of thinking about the problem, but he was very reticent to take credit for that. And I think his ability to have that sort of vision and be able to really push forward what we're trying to do with what to many might seem a very trivial observation, but actually I think really transformed the way that we thought about dislocation motion in a way that was actually very useful, has really been pretty profound. And I really thank you for that. That's just one of many contributions. I had a number of other mentors. I worked for John Brodman. I was his graduate student. And I did have as well the great joy of working with Ted Jabal. Bob, I think you're in the audience, so I tried to find the most flattering picture of you that I possibly could. That's actually from Bob's graduation at Cambridge. It turns out there was a testimonial to Bob in the ultramicroscopy journal where somebody had dug that up that I was able to find. But just a couple more anecdotes. John Brodman is still an interesting character. One of the things, being an electron microscopist, that I tended to do was work nights because the resolution was better. It was not infrequent that I would leave at 4 o'clock in the morning and let John into the building. So different people have different work styles. Those of us who worked with Ted Jabal very quickly learned that if you had a meeting, noon meeting with Ted, bring food. Because Ted never broke for lunch, and if you brought a sandwich, he would eat half of it. Breaking bread makes the world go around. And one of the stories that I actually like to tell, and this is where it's true but probably not accurate, is working in the electron microscopy labs at that point in time in the Peterson building. Bob would occasionally mosey in, and I remember him coming into the microscopy lab at one point, just sort of doing that thing that Bob does, sort of rolling back on his balls of his feet and saying, when I was a graduate student, when I was a postdoc, when I was doing work over at Berkeley, we didn't have video cameras. We didn't have all these fancy tools that you have on microscopes now. We had to take an image, we would go and develop the plate, we would then take that and look at it and see if the lenses were aligned correctly, and then we would go back and realign the lenses, sometimes with Allen wrenches, and then we would take another image. And you're sitting here with potentiometers and a video camera, and you can just do it in a few minutes. You have no idea what electron microscopy is actually like. And there's some truth to that if you actually stop and think about it. So what Bob was really pointing to was a transition over time that I think has really manifested itself in a number of different technologies, where in the early days, it really is a specialist technique where it takes a lot of dedication and expertise and discipline to be able to get the tool to work, just using this particular example. And over time, what happens if a characterization technique or some other method is actually effective, it becomes more of a black box. We figure out how to make that accessible to people who are non-experts. And over the course of the last 30 years, this has really happened in electron microscopy. The Colin-Ophuses of the world are actually quite rare now. And the reason for that is because in many cases, you can take a TEM or an SEM, you push a button and the computer aligns it. You're not even doing it yourself. So maintaining that accessibility to a broad community and balancing that against the creativity that, for instance, Colin is showing by really pushing the state of the art and really pushing the forefront of what we can do is one of the balances that we're always fighting as a scientific and technical community, where you got to get that balance right. It's easy for it to tip too far versus one versus the other. So that was one of the things I really took away from my time with Bob. Turning back to the story I promised I'd tell, one of the things that was interesting talking to George when I went to visit it is it turned out that when George was at Stanford, he had a number of reprobate friends who included Paul Foose, Steve Latterman, Sean Brennan, and ultimately Brian Stevenson. And they had worked together. Several of them had been in the same research group. Several of them actually lived in the same house or apartment. And I discovered when I moved to Argonne in 1998, actually was joining work that Brian Stevenson was doing on metal organic chemical vapor deposition, originally on nitrides, but ultimately on oxide thin films as well. Paul Foose eventually moved from Bell Labs, actually from AT&T at that point, back to Argonne in about 2002, if I'm not mistaken, sometime around that time period. And we were working on these experiments using a deposition system, which is shown right here in the middle, which had turned out to be, it was kind of the axe where you had changed the head and the handle, but we still called it the same deposition system. It was originally built by Dave Kisker, Sean and Paul and others, and was run at the PEP beamline just before the Loma Prieta earthquake. And the reason that this thing wound up at Stanford is after the Loma Prieta quake, PEP was never reopened as an undulator source, so Brian was eventually able to get the system shipped to Argonne to rebuild there and start to use for experiments. It was actually very interesting because there was all this equipment with this guy's name on it, Foos, who I'd never met. And then one day he showed up, and I think that was a good thing. The interesting generation of this is then if you move about 10, 15 years further, that system was rebuilt again. The remaining components that had been part of the SSRL PEP installation, the goniometers and the exhaust cabinet were eventually replaced with something of a more modern hexapod to allow better range of motion. And what this really shows is an evolution where there was the first glimmering of being able to look at surfaces in situ in a complex metal organic chemical vapor deposition environment where there was a glimmer that we could do cool things. That then moved to the advanced photon source that actually demonstrated that that would work, and I'll show some of the highlights that Paul referred to from that work in a second. Really looking at macroscopic surfaces. The footprint of the beam on the surface was a few millimeters by a few millimeters. So although we were getting atomic resolution detail in the out of plane direction and atomic resolution detail from scattering in the in plane direction, again by rotating and other tricks, we weren't really doing nanoscience in the way that we can do it now with coherent beams, with X-rays in exactly the same way that Colin is describing it with electrons earlier. And that's really what the third generation instrument is targeted at, being able to use the increasing coherence that we have with light sources right now and use that to get not macroscopic information on microscopic phenomena, but microscopic information on microscopic phenomena. So that ability to start to probe short range order, to probe crystalline order at short wavelengths, short length scales, is really the forefront of what we're trying to do with many of the scattering probes that we're using right now. Just to hit a few highlights of this, it really was a fun time. We had a lot of work that we were doing on essentially establishing clean surfaces, using that as a baseline by which we could then produce films that were reproducible and then whose condition were known, and then looking at phase transitions in these films in a way which is actually quite astounding. And the fact that we were able to measure TCs as high as a couple hundred degrees C above the bulk temperature because of the strain states of these films was really remarkable at this time. And really set the stage for the fact that this particular type of collective phenomena persisted down to length scales that at that point in time people thought were impossible. Now finally, there's another piece of the thread through Oak Ridge National Laboratory, through Argonne National Lab, that I think is also crucially important and really gets back to the way that Brian Stevenson in particular has been a real leader for the community. So Brian, of course, got his PhD from R.D. Bienenstock's group. He followed the group of folks who were over here. His PhD was in 1983. He did a postdoc at Rice, then bringing back that connection again before he eventually moved to IBM, and then ultimately to Argonne National Laboratory. And it turned out that Brian was the director of the advanced photon source prior to me taking over. And in about 2012 or so, Brian realized the design for the upgrade project for the APS was the wrong design. And through a Herculean effort of not just Brian, but many people at the APS, they redesigned the magnetic lattice for the facility and dramatically shifted the future of the APS from what had originally been basically a beamline upgrade to something that replaced the storage ring with something called the multi-bin achromat lattice that increased the X-ray brightness and the coherent flux by somewhere, depending on the energy you're looking at, somewhere around a factor of 500. And that is transformational in what you can do with that. So Brian had his period as the APS director. He stepped aside and I took on after him. And just to show you the duration of some of these events, the APS upgrade just completed its final paperwork for what the Department of Energy calls completion of a project, CD4, in January and February this year. So it's important to remember that even though the speed of innovation is continuing to improve, the timeline of some of the things that we're working on really do take multiple decades to come to fruition. So the first glimmers of the APS upgrade were actually in the early 2000s, coming online in 2025. One of the things that Jeff Wadsworth started at Oak Ridge was actually the US involvement in the ITER project in France, the tokamak experiment in France. That was in the early 2000s. We expect that facility to come online early 2023s, or the early 2030s. So it's important to remember that we've got to keep this sustained focus on science and technology, and that science and technology has a rhythm that is interesting. We have very rapid advances like we see in artificial intelligence that have been brewing for decades, where all of a sudden there's a breakthrough that transforms the way that we do science and technology. And there are other things that we do that really are decades of dedicated effort that ultimately are important for the community, that move the needle in ways that are almost glacial. And having a system that allows us to do both and allows us to move forward and actually meet the needs that we have as a world is part of the challenge that everybody in this room is taking on. And it's a fun one. So I'll stop there.
Great presentation. I'm also a 93 graduate. So good to see you. Question, you talked about the data centers and things that you guys are working on, right? That's the hottest topic right now. The number of data centers proliferating like crazy. We don't have enough energy to power them.
Absolutely.
Right? And every idea that we're hearing is wackier than the next. Orbiting in space, all kinds of stuff. But the question I had specifically is that a lot of buzz that I hear is that small nuclear reactors are the answer and they're going to solve all problems. Is that true?
If you listen to the companies that are building small modular reactors, yes. There are a few things I'd like to unpack there. So the largest data center in the US right now is still Colossus, the Colossus installation, which, given Elon Musk's sense of humor, he actually calls macro hard is the name of the facility. You can guess what that was targeted at. And that's only actually 700 megawatts right now, scaling out. If you look at data center capacity, right now it's about 4% of electricity use in the United States, and about the next 5 years it's expected to get to about 10% of electricity use. So I think there are two things that are going on in parallel. Obviously, there's an international race to try and build the infrastructure that really is necessary to do all the AI that everybody wants to. But a lot of it is actually boat building. We're going to build a data center that's 2 gigawatts, and that's going to shut out all my competition. And you're absolutely right. We can't actually afford the energy to run all these data centers. There are lots of other energy drivers. Yi can probably talk about this better than I can. But I think what we're seeing right now is not only in the Tennessee region, but around the US, electricity usage had been flat for about 2 decades. And we were trying to understand how to start to take carbon generating sources out of the electricity generation business in a regime in which electricity utilization was flat. And in that case, SMRs actually make sense. I have a friend who's the director of Idaho National Laboratory. The problem with SMRs, John Wagner, who's the director, likes to point out is small modular reactors currently are not smaller modular. As you start to look at it and look at the complexity of actually building any reactor at scale, and if you look at, rather than a flat demand, an increasing load demand, people are starting to move back to full-size plants again as part of the mix. So I think it really is one of those cases where we have to work on everything. We need solar, we need wind, we need geothermal, we need hydro, we need small modular reactors, which have a role in the ecosystem, but we got to build full-size reactors, otherwise we just can't keep up.
Please join me in thanking Stephen. I'd next like to invite my colleague, Yi Shui, the Fortinet Founders Professor of Material Science and Engineering, to introduce our next speaker.
Thank you, Sarah. It's my great honor to introduce our keynote today, Professor Moungi Bawendi from MIT. Let me give you a formal version of introduction first. Moungi got his undergraduate degree at Harvard. After that, moving to University of Chicago to do his PhD. Following his PhD, he moved to Bell Lab as a postdoc working with Louis Bruce. That was the time it's really starting the whole new field of quantum dot. Moungi is a co-founder of this field. After 2 years postdoc at Bell Lab, he joined MIT as a faculty member, built his independent career, and he's very well known as a person, as a co-founder of the field, developed these very high-quality quantum dot materials by developing its synthesis. He's also very well known for his very deep study on light-matter interaction based on quantum dot materials. Over a few decades, he also explored the applications of quantum dot for photodetectors, for solar cells, for labeling used in biology. So it's really broad range of applications. For his pioneering work, he was awarded the Nobel Prize in Chemistry 3 years ago. So this is the official introduction of Moungi. 2005, I joined the Stanford faculty. I really like what Steve Jobs said in the commencement speech. Life is about connecting the dots. Now let me connect a few dots right here between Moungi and Bill Nix. So in 1998, I went to graduate school at Harvard, starting to work for Charles Lieber and nanoscience. I started to read a lot of papers. And then turned out to be a lot of papers I read is Moungi's papers. Learning about what happened in nanoscience, what's possible. And Harvard MIT has this joint physical chemistry seminar. So I will go to MIT this week. Next week, maybe Moungi will come to Harvard to listen to the seminar. This rotate back and forth. That was the time I started to, as a graduate student, sitting in the audience, seeing Moungi sitting somewhere in the front row. That's watching Moungi remotely since 1998. Moungi just didn't know about this. So all our faculty members remember that our students are watching us, just like I watch Moungi. So things fast forward. 2002, I graduated. I went to UC Berkeley, working for Paul Alivasatos as a postdoc fellow. Paul, actually, and Moungi, two of you work together at Louis Bruce's lab. So suddenly, Moungi, you become my academia uncle. So that's the first thought I want to put in. And in 2005, I joined the Stanford faculty. And remembering interviewing the faculty, with faculty right here, it's such a great environment. Shortly after my visit, so I joined in the faculty and decided to work on energy instead of initial proposed ideas. So this environment has been so supportive. I started a battery research program. As soon as I started to work on batteries, I found out there's a lot of mechanical issues. Chemistry and mechanical coming together. I really know very little about mechanical property. Bill Nix, David Barnett, Rainer Dauska are really the three faculties teaching me about mechanics. I started to form very close collaboration with Bill. Bill, I still remember you, me, and David Barnett, right, the SB twist, and 9 p.m., 10 p.m., we email each other back and forth, how do we understand SB twist? That just is incredible experience in collaboration with Bill. And my mechanical property, the knowledge advanced. And then one year, I hired a high school student into my group. Oftentimes, every year, I hire a few as my outreach to the younger generation, encourage them to go into science. There's a smart young student coming into my lab. His name is Oliver Nix. After a couple of summers in my lab, he actually majored in science. And a few years later, he told me he was joining MIT as a PhD student. And then recently I learned he worked for Moungi Bawendi. So Oliver Nix is the grandson of Bill Nix. Now I'm connecting all the dots together. Moungi Bawendi, Bill Nix, Oliver Nix, E-Trade, right? So we are all connected. So as Steve Jobs said, life is about connecting the dots. I also see for our department alumni right here, you have your version of connecting the dots at Stanford Materials Science right here. Today, it's a great honor. Moungi, I'd like to welcome you to the stage to give the keynote address. Let's welcome Moungi.
Well, it's my pleasure to be here today, and especially to honor Bill Nix in this seminar series. And as Yi pointed out, I've had a long relationship with Yi, looking far away, and been so impressed with the work going on in his career in the nanoscience area. And then it's really been my pleasure to have Oliver work with me as I'm his co-advisor, and learning about fields that are adjacent to me, and having him in the lab has been an amazing experience for me and for the lab as well. And I'm sure he, as he told me, he's inspired by his grandfather, and I can see after meeting Bill Nix, I can see the inspiration and learning about the science that was done here. It's really an amazing place to be. And you know, science is a social endeavor. We all work together, we're all connected together, and it's actually a much smaller community than one realizes. I go throughout the world and everybody's connected to each other in some, you know, it's 6 degrees of separation. You can find your relationship with anybody in science, it seems, in so many different fields. But in any case, I'm really happy to be here today on this occasion. And the seminar, or the talk I'm going to give to you today, is really an origin story of the field of quantum dots. And embedded in that is really an origin story of the field of nanoscience, because the dots that I'm going to be talking about really were one of the first nano-engineered materials where we could really control the properties of matter at the nanoscale with essentially near atomic precision. And I was really impressed with the talks that I heard this morning, Jennifer's talk and Colin's talk, because you can see the incredible evolution of this field of nanoscience in the applications and of the field through Jennifer's talks, and also through the ability to be able to image what we're actually making. Because as you'll see, electron microscopy really has been a tool that has been extraordinarily important to this field, because if you can't really see what you're making, it becomes really hard. And the better we can see, the easier it gets to really engineer these materials in a way that we can use them and study not only their applications, but also their fundamental science. So my talk is titled From Curiosity to Technology, because it's also a talk about the timeline of discoveries from initial ideas to having them be throughout our society. It's a long timeline. It's a timeline that really hasn't changed that much. I mean, as was pointed out, you know, we feel like AI just burst on the scene, but it's been brewing around for a long, long time. Machine learning is something that we used in my lab already 10 years ago. You know, and it's the sudden explosion has to do with the confluence of different technologies coming together, computing power, theoretical ideas in machine learning coming together, neural network ideas, all that stuff, you know, suddenly merging together. And the story of quantum dots is a little bit like that as well, where there's a period of fundamental discoveries that feel very theoretical, very curiosity-like, very fundamental. And then when the field gets advanced enough, and it doesn't happen to every field, it's hard to predict when this is going to happen as well, you start to connect the dots to other fields. And when you connect these dots, you realize that what you're doing is really can be applied to other areas, and everything begins to feed on each other, and then you have this explosive growth of people working in the area, and adjacent fields being created, et cetera. So let me, without further ado, go ahead and tell you the story, which is really my story in some ways. So let me start by telling you a little bit about myself. Colin mentioned he was also an immigrant. I am as well. I was born in Paris. My parents are French and Tunisian. My father's Tunisian, my mother is France. Tunisia, for some of you who may not know, is a little country in North Africa. And as a result, as a child, I moved back and forth between Europe and North Africa, between two different cultures. Quickly became aware that I was neither part of one culture nor the other culture, and understood that I was kind of an outsider everywhere I went. And that's a feeling that kept at me throughout my life. And it's also a feeling that I think by having to understand how to deal with that, also influenced my science. Because I wasn't afraid to be an outsider when I first started working on what I was working on, because, hey, you know, that's the way I was my entire life. Eventually, we ended up in Nice in southern France, and my father ended up at Purdue University. He was a mathematician. You know, when you're in between, this was 1971, when at the time, you know, North Africans were not particularly welcome in France. I can't say that they're particularly welcome now either. And in Tunisia, there was just no infrastructure to do science. And I can't say that there is infrastructure today. It hasn't changed that much. But the US was an inviting place where when we arrived in West Lafayette, Indiana, I would say, you know, my father's from Tunisia. And they would say, oh, how interesting. Tanzania? And, you know, they had never heard of Tunisia before. So we ended up in West Lafayette for a few years and ended up back in Paris. Oh, this is me as a 10-year-old in Orly, not speaking a word of English, getting ready to go to Chicago on this flight from Paris to Chicago. And when I first arrived, I hadn't believed my mother that people didn't speak French in the US. You know, how can you not speak French? People speak French all over the world, like in Tunisia, for instance. I never had to learn Arabic, you know. My first experience arriving in this rental house we had, and there was a kid across the street coming to play with me and started to speak in English, and I had no idea what he was saying. You know, I was a complete outsider again. West Lafayette is the home of Purdue University, a wonderful place. And Purdue, if you replace the first U with an E, in French, it spells perdu, which was completely appropriate for me. I was completely lost. But I did okay. You know, we went back to Paris for a year in high school and back to West Lafayette, so I really was between continents there as well. Then when it came time to apply for college, my parents, being outsiders as I was, had no idea what the process was in the US, you know. And so I applied to every place. I applied to Harvard and Stanford and Princeton and all these places that their friends told them I should apply to. And, you know, I got into a lot of places, including Stanford and Harvard. And because we didn't know anything, you know, we asked our friends, you know, where should he go? And we said, oh, he got into Harvard. He should go into Harvard. You know, forget about Stanford. But Stanford had a different reputation back there in the, you know, 1,000 years ago than it does today. Maybe I would have made a different choice today. In any case, then, you know, at Harvard, this was 1978. Harvard was still a place that was dominated by East Coast private school kids. And I was from the Midwest, you know, it's like, okay, so back to being an outsider. And I was very intimidated by all the students who had gone to math summer camps and physics camps and all this stuff, and I hadn't done any of that, and I thought there's no way that I'm going to be able to take any of these advanced courses because they all know all this stuff already. But chemistry was something that I had a great teacher at in high school, and I felt comfortable taking you know, chemistry as my first class, and I loved the subject. And then I took my first exam in chemistry and promptly got the lowest grade in the class, you know, 20 out of 100. That was an interesting setback, and I realized then that I didn't know how to study. that I knew the material but had no idea how to take an exam. And so I had to learn how to take an exam really quickly. And after that, everything was fine. But you know, science is a series of failures, and this is certainly one of my first failures. After Harvard, I went to the University of Chicago, influenced by my father, who's a mathematician. I decided to be a theorist, pen and paper theorist. I was a polymer theorist at the time, doing statistical mechanics and computing thermodynamic quantities. I had never really understood thermodynamics. I could do the math, and I could do all the statistical mechanics part. But then I calculated these thermodynamic quantities. I really didn't really understand what they meant. And that was a problem. I felt like I didn't have any intuition as to what I was doing. I could do the mechanical part of the science, but not the intuitive part. And I realized that, and I realized this wasn't going to happen. So I switched to being an experimentalist halfway through. I became a small molecule experimentalist, a spectroscopist looking at the low vibrational spectra, the infrared spectroscopy of small molecules, small ions that are interesting for astrophysical observations, like H3+. H3+ is the smallest polyatomic molecule you can think of. It's got 3 protons and 2 electrons. It turns out to be a very complicated molecule to study its structure and its dynamics because it's extremely floppy, because the 2 electrons aren't quite enough to keep the 3 protons sort of rigid, so they do all sorts of motions, and so the spectrum, the vibrational spectrum is extremely non-harmonic. And so that was what I did as a graduate student, my second part of my graduate student career, and that's where I fell in love with quantum mechanics. Quantum mechanics is this amazing way of looking at the world. It's completely non-intuitive, and it's very magical, and it's an amazing thing. And so when I had the opportunity to go to Bell Labs for a summer, I had an AT&T fellowship, I went, and Lou Bruce chose me. Life is a series of accidents, right, of opportunities and accidents, and this was an accidental meeting between me and Louis Bruce through this AT&T fellowship, and his looking at me on paper and deciding, oh, let's try him as a summer student. And I went there, and I met Paul Avasados and Louis and the whole team at Bell Labs, Mike Steigerwald and Tim Harris and all the people that were, and experienced the atmosphere, the amazing atmosphere of Bell Laboratories at the time, which was this incredibly interdisciplinary place, which was rich in equipment and not so rich in human capital, because everybody had to work together. It forced people to work together and also forced people to think really hard about the kind of projects they were going to do, because they were going to be all by themselves with maybe a postdoc doing it and working together. And it forced them to think about, am I working on something that's really important or not? We have this luxury in academia that we have graduate students, and some of us have more than others. And so, if we work on something that maybe not as important as something else, well, it's okay, because we have many graduate students. And if some of the things aren't going to pan out, well, we have other graduate students, But at Bell Labs, you couldn't think that way. You had to be really sure that that one problem you were working on was really important. And, and I learned that. I learned that from Louis. So after Bell Labs, after Chicago, I went to Bell Labs as a postdoc because I was hooked on quantum mechanics, and I was hooked on these little particles that he had discovered in the early 1980s, these quantum dots, these nanoparticles. We actually weren't calling them quantum dots in 1990 yet. The term quantum dot really was invented by physicists a little later. The physicists have a great way of finding names for things that really stick. They have a way of doing that. We just called them little particles, and that didn't work. That wasn't as good as quantum dots. So I went back to Bell Labs and learn again more about. When I came to Bell Labs, Louis told me, oh yeah, cadmium selenide quantum dots or nanoparticles. That's done. You're going to be working on 3.5. So I came in and very quickly realized that wasn't the case. The 2.6 quantum dots were not done at all. And I'll tell you the story of that in a little bit. After that, I came to MIT, where I started my career in 1990, and where I am still today. So let me start by telling you what a quantum dot is, for some of you that may be a little bit outside the field before I tell you the inception story of the quantum dots. So a quantum dot is a small particle of a semiconductor. It's nano-sized. It's got a few thousands to tens of thousands of atoms in it. And when you put an electron in a quantum dot, it shows quantum effects. And the importance of imaging in the field can't be overstated. You can't see them with a light microscope. You have to use an electron microscope. And without an electron microscope, you really don't know what you're making. You can infer it using maybe small angle X-ray, but if you can't see it, it becomes really tough. And so this is one of my students working on one of these early microscopes that still has knobs on it. I love knobs. And when you have a good sample, then you can actually see that you're making particles that are all about the same, and you can measure the size and all that stuff. And the visual aspect of it in the microscope is just incredible, because now you can go back and forth and really optimize things in a way that you couldn't do otherwise. Okay, what about the quantum effect? The quantum effect is because the electrons are now stuck in a very tiny box. And what do I mean by that? If you think of an electron in a wire, the electron behaves as a particle in a wire. You know, it carries electricity, carries charge, etc. But if you take that electron and you stuff it in a nanobox, a box that's on the order of a few nanometers or 10 nanometers in a semiconductor, for instance, where the electron has a slightly less effective mass than in a wire, then the electron doesn't behave as a particle anymore. It behaves like a wave. It becomes a quantum mechanical object. And those quantum mechanical properties are then what drive the properties of that electron and what we observe then when we do experiments. And that nanobox is a quantum dot, right? It's our particles. Our particles behave as a box for this electron, which then acts as a wave. Now, the properties of wave are inherent to quantum mechanics. It's what makes quantum mechanics what it is. And the particle-wave duality is one of the basic tenets of quantum mechanics. And so, but we understand waves because waves are all around us, and we understand the properties of waves. We know, for instance, that cavities, like the quantum dot and waves, interact in ways where the property of the wave depends on the size of the cavity. So if you have a look at sound waves, a flute has a much lower pitch than a piccolo. You know, we hear that. We hear the size of the cavity with our ears, right? The same thing for organ pipes. If you have the little pipes have a higher sound, higher pitch than the big pipes, and the medium pipes are somewhere in between. We hear the cavity coming out of the pipes through the sound waves. The same thing happens then with the nanoboxes with our electrons. So when we put an electron in a nanobox, somehow put an electron in a nanobox, then the electron the nanobox is the cavity, the electron is a wave, and the size of the wave depends on the cavity. How do we put an electron in a nanobox like this, where there are already lots of electrons in there, because this is a material that has atoms that are actually pretty heavy, like cadmium or selenium or indium or phosphorus or things like this. But as you know, in a semiconductor, you can release one of the electrons that's bound to an atom with light. You can inject energy into the material, create what's called an exciton, release that electron. That electron becomes a free electron. It's a free carrier, right? And it's confined into that box. So that's how we create that one electron in the box, you know, by shining light on it. Now we have that one electron in the box. It's this wave. And eventually it finds where it came from. It finds the hole and emits a photon. It releases the energy of the wave as light. The energy of the wave as light is kind of like the sound that we hear from the organ pipes, right? So instead of hearing the size of the cavity, now we see the size of the cavity, with the small cavities being bluer than the big cavities. And the material that we're studied, which has become the hydrogen atom of the field, is cadmium selenide. And the reason for that is not a coincidence. It's because the energy of the light that comes out is in the visible. So we actually see it with our eyes. We don't have to go to a spectrometer. It's not in the infrared or in the UV where we'd have to do measurements back and forth. We can actually do the experiments, we can make the material in real time and optimize in real time because our eye becomes the detector. And that became incredibly useful for optimization. We don't have to go back and forth to the microscope. We can optimize first with our eyes and then finish the optimization with more sophisticated tools. And so the outcome of making this then is particles that emit light, that are emissive, an emissive material where the color depends on the size of the particle. In 2023, then I was in Stockholm with Louis Bruce, my mentor, and Alexei Ekimov. And Louis and Alexei are the two in this Nobel Prize trio that are the discoverers of the effect. Let me tell you a little bit about their story. Both of them came to the discovery of quantum dots through really some intrinsic curiosity about the world around them. Alexei was working in Leningrad, which is now St. Petersburg, in the late 1970s, early 80s. And his interest, and he was a young man at the time, his interest was in understanding what gave color to stained glass windows. And it was already known that the red was due to small gold particles, that if you take gold and you chop it up into little tiny pieces, and this was known in the late 1800s already, 1700s actually. Faraday knew that, discovered that, and you can see samples in the Cavendish laboratory. And the Greeks knew how to make red color from gold. But what about the other colors, right? And so his interest then was to take glass and incorporate different elements in it to grow crystals in the glass and see what happened. And what he discovered was that depending on the processing conditions of the glass, he ended up with different colors. And by using small angle diffraction, he was able then to correlate the size of the particles that were growing in these glasses to the spectra that he was measuring in the lab. And he was around physicists at the time that were working on quantum wells, thin sheets of semiconductors, which had been already discovered and studied, which were known to be, the properties were known to be due to quantum confinement, to the confinement of electrons in one dimension, as opposed to the 3 dimensions of a quantum dot. And so he understood what he was observing was due to this quantum effect that I described, and he wrote a paper that came out in the Russian literature in 1981 by the title Quantum Size Effect, the first 3 words, in 3-dimensional microscopic semiconductor crystals, as you can read. This was in Russian. This was before the fall of the Berlin Wall, and this paper was completely unavailable in the Western literature. Lewis was working at Bell Labs, and this was the energy crisis of, under the Carter administration, OPEC, embargo, etc. And as today, there was a lot of push to try to see how to make fuels from sunlight. And one of the things that was proposed that you use semiconductors to harvest sunlight and then do electrochemistry to create fuel at the interface. You take an electron from the semiconductor, you do electrochemistry, you create some fuel that you garner. And so he was interested in how fast does that electron move across that interface from the semiconductor to the electrolyte in solution, to the solution. And there was a new tool that had invented in the 1970s called a picosecond laser. And that tool was then able to create pulses that were short enough to be able to see or to measure the speed at which that electron went across that interface. So, tools are really important. So the tool then is what drove his research, his initial research. Just like the microscope, the electron microscope is then eventually what allowed us to see the quantum dots. So using this new tool, he started measuring the Raman properties of the particles, of the molecules in solution as they changed, as the electron, as they became charged from the electron that was created in the semiconductor. And because it's an interface process, it comes from the surface, in order to optimize your signal to noise, you want to make the surface to volume ratio as high as possible, right? And how do you do that? Well, you take your semiconductor and you chop it up in smaller and smaller pieces. You get your surface to volume ratio as high as you can, and he ended up in the nanosize regime using recipes that had been initially developed in Germany by the group of Henglein, who was a radiochemist, who was also interested in similar things. And he was using cadmium sulfide, and he realized that if he let his samples sit, they changed colors. And sometimes even from the get-go, they didn't always come out the same way. And at the time, these were made in water at dry ice temperature, where you arrested the growth of the crystal growth, at low temperature. And because he was at Bell Labs, which was full of wonderful tools, they had an electron microscope. And so he was able then to use the electron microscope to correlate the size of the particles that he was using, the growth of the particle as they set on the bench with the color that he was seeing, that he was measuring. And he wrote a paper in 1983, came out in 1983, with the first 3 words, quantum size effect. And the other words in the title have to do with why he was doing this, the redox potential, et cetera, using resonance Raman to try to measure the electron, at the surface, on the molecules that were at the surface. But he explained the effect in exactly the same way that Ekimov had explained it in Russian in 1981. Science was ready for that discovery, ready because of the confluence of the physicists working on quantum wells and the tools that were being created to both study these things as well as to create them. The English translation came out around the same time in 1983 of the Ekimov's paper, and Lewis eventually found this paper and wrote a letter to Alexei that they were working on the same thing, and they should meet or something, or work together, or compare notes. And that letter led to was diverted on its way to Alexei by the KGB, and that led to, well, it didn't lead to an international incident, but the KGB was highly interested in why an American scientist would send a letter about this to a Soviet scientist, but quickly realized that this was completely useless and harmless. Okay, where do I come in? I come in the synthesis part, and through a paper that we wrote in 1993. This was my first paper at MIT, and I'm going to tell you that story now. Okay, so when I came to MIT, when I was at Bell Labs, the way that the particles were made were in water. And this was a discovery, a method that Paul Avesados had worked on with Mike Steigerwald, who was an inorganic chemist at Bell Labs, with Bruce, with Lewis. And basically the idea was to make little reactors with soap, right? You make inverse micelles. You make little tiny reactors in an organic solvent, and inside each little bubble of soap is a little bit of water, and that limits the number of ions that are in that little bubble, So you have a few ions of cadmium, you introduce some selenium, in this case through bis trimethylcytyl selenide, which was what Steigerwald knew, all this inorganic chemistry that none of us knew, and that way you made little cadmium selenide crystals inside the water at room temperature. And these are my transparencies from 1990. When you get a Nobel Prize, you go back through archives and you try to find things. And so you can see that I drew this particle as being completely misshapen, surrounded by phenyl groups, which was also the genius of Mike Steigerwald of realizing that the surface was still chemically active, that you could arrest the surface and then use these phenyl groups to be able to redisperse these things in organic solvents and begin to anneal them. But basically, these were very defective particles, huge size distribution, very misshapen, and the fluorescence that we use today that is applied was non-existent. They were basically emitting white light in very, very low quantity. So when I came to Bell Labs, there was a project that there was a an observation that Mike Steigerwald had made, which was that when you took these particles, these smaller particles, and you put them in pyridine and annealed them to try to get them to be better crystalline, the spectrum that would come out would always come out the same. It was this very sharp absorption at 410 nanometers. And they wondered, you know, what does that due to? Is this because we're creating little clusters that are all the same, they all have the same shape and everything, maybe we can crystallize them, or maybe it's a charge transfer band. You know, the electron is sitting in the particle and the hole is sitting in the pyridine, and we're seeing the transition seeing that transition. That was my job to figure that out, and I had never done chemistry in my life before then. And you know, so Mike Steigerwald told me, yeah, you know, pyridine is a Lewis base, and I kind of stared at him and said, a Lewis base, what's a Lewis base? You know, I learned acid base as a freshman in chemistry in Harvard, but somehow I missed the Lewis base part of it. So I asked him what's a Lewis base. He explained it to me. Then I went to around his lab, and I found all the little bottles of Lewis bases in his lab, all the phosphines and amines and pyridines and everything that was there, and started cooking these little particles and these Lewis bases. And I found this bottle of tri-butylphosphine that was, you know, I had probably 30 little containers, you know, cooking at any time. This was. We didn't use that word, but this was combinatorial chemistry, you know, manual combinatorial chemistry with artificial intelligence here. But so I found this little bottle of phosphine that the particles came out, and they came out all the same. First of all, it grew a little bit, and then I went and took some powder X-ray, and the crystallinity had increased tremendously, better than we had ever seen before. And now there was a little bit of light also that was coming out in fluorescence. It was just this magic thing. And if you notice on my transparency, there's the word magic sitting up there from 1990, because I had no idea what was happening. And on top of that, it didn't happen every time. It only happened once in a while. But it happened enough that I was able to do some physics with it. And they always came out the same size, always around 3 nanometers or so. So then I came to MIT, and I proposed that I was going to do this. And my proposal, really my curiosity-driven proposal, was to understand the change in properties as you go from atoms to the bulk. You know, this transition from atomic properties to bulk properties as we go through the nano size regime. What's going to happen there? You know, how do we go from an atom of silicon that looks nothing like a bulk silicon, and what happens in between? And I needed to make samples, and I had proposed to make samples based on this magical discovery from Bell Labs. And so I came to Bell Labs in 1990, and quickly was unable to reproduce anything that I had done at Bell Labs. The magic of New Jersey that happened every now and then no longer happened in Cambridge at MIT. I had two incredible graduate students that joined my lab in September of 1990, Chris Murray and David Norris. Chris is now at Penn, and David is at ETH in Zurich. And they couldn't reproduce it either. And so we had to reinvent the synthesis based on what I had learned at Bell Labs. What did I learn to get to this magical thing? What I had learned was that you needed high temperature. I had learned a lot of things that are really common sense. And the common sense part, in retrospect, sounds really easy. And the devil is always in the details, right? Making it work. So I had learned that you needed high temperature. You couldn't do it in water because the surface of a semiconductor in water just wants to oxidize. It had to be under inert atmosphere. It had to be organometallic chemistry. And we had to separate nucleation and growth somehow, which I had done at Bell Labs by making little particles in water and annealing them in these phosphines. We had to combine everything in one go, and we couldn't use tributylphosphine for two reasons. One, it wasn't, the boiling temperature was too low. We wanted to go to higher temperatures. And second, it was way too expensive. And I was burning through my startup money buying little bottles of tribrylphosphine, which just were not working. But we discovered that trioctylphosphine, with a much higher boiling point, it happens to be a solid, the oxide is a solid at room temperature, tribrylphosphine oxide, is used for the extraction of uranium. And it's made in the train load, as opposed to little bottles. And it's really, really cheap. And so we went to that, and that turned out to be really, really important, because now we had high temperature, and we were able then to create this, what is called now the hot injection method. So this is basically, we take our metal organic precursors that contain our cadmium and selenium, or whatever elements you want to put in there. We've got a solvent, a coordinating solvent where we use this octylphosphine compounds. High temperature, 300, basically as high as an organic chemistry can go. You know, organic solvents don't really go much higher than about 375, 400 degrees Celsius before they start falling apart. So we basically push the limit of organic chemistry, organic solvents. And it worked. It worked. We were able then to create a size series of these particles. You know, Chris did an incredible job of learning polymer methods also to be able to purify these things. And now I have a laser printer. You know, it's 1992 or so. Laser printers are available. And these transparencies have both handwritten and typed things. And you can see it, you know, the really simple idea. High temperature, uniform surfaces, all the same molecule, the nucleation and growth, the rapid injection, and then the extraction as a powder based on what Steigerwald had realized using the aqueous thing. And this hot injection, there you go here at a small scale. This is from a YouTube video from 2010 from one of my students. There's a great soundtrack, you know, if you want to watch it. I just go for the soundtrack myself. The quick color change, creating the little seeds of the little boxes, and you can see the creation of the one electron in the box giving you this orange color or this red color. And then if we wait like 20 minutes, so we've waited about 20 minutes, now it looks dark, you know, really black. They're much bigger boxes. Still extracting samples out of it. And then if you take if you dilute it out, you can see the color differences, which is obscured by the concentration in the main flask. And then if you shine a UV light on it, then you can see the effect of the cavity on that one electron wave, giving you different colors coming out from that exciton. So then we wrote a paper. You know, I started in July 1990, and this is my first paper that came out in 1993, about 3 years later. And today, if we had an assistant professor that didn't write any papers for 3 years, you know, we'd be a little worried, right? Be a little worried. But in any case, the paper has underlined a few things that, in retrospect, were really important to make it into technology. It was simple, and it was a single reaction. It gave monodispersity. We have something called psi-selective precipitation, which is a fancy name for purification of a nanomaterial based on essentially purification of polymers. Simple and single reaction are important, as you know, because the cost of anything depends on how many steps you need to do to make the thing. It doesn't matter what it is. If it's a car, if it's a pharmaceutical compound, the cost of making it depends. If it's a chip, you know, NVIDIA chip is much more complicated to make. So many more steps than a simple chip, right? So simple and single reaction meant that if you were to scale it up, it would end up being inexpensive. The process by which today quantum dots are made at scale is very, very similar to the way we made it in that video. Hot injection, except now instead of being a 50 milliliter flask, it's a 3,000 liter container, steel container. Everything is automated from start to finish, including the purification. And then at the end of the process is a barrel of quantum dots comes out of the process that emit light with 100% quantum efficiency. The labor costs are gone. You know, the labor cost is a person basically pressing a button to start the process. Everything is based on the cost of the materials. When we first started thinking about applications as a field, there was somebody wrote a news and views in Nature saying how this could never work as a technology or because the cost was going to be too much. And they estimated the cost was going to be $10,000 a gram. But what they forgot is that when you scale things, costs start to plummet very quickly. And if you scale at high enough dimensions, quantities, the cost essentially becomes the cost of the energy required and the cost of the elements that you put in there. Everything else goes away pretty much. You have the initial capex of building the process, but after that, the operating costs are energy and the cost of the elements because you can then control your supply chain and your costs can really go down. And today, the cost of making the dots is about $1 a gram. It's pretty much close, maybe 50 cents a gram. It's pretty close to the cost of the energy plus the elements. So what did we see that we didn't see before? Well, we saw what I call, in here, the defined transitions. They are basically the harmonics of the wave that are in the cavity. We could start to see the harmonics, just like when you hear the flute, you hear all the harmonics, or guitar string, pluck a guitar string. And as a spectroscopist, the harmonics was really interesting to you. Those are the excited states, right? And then we saw light coming out that was very uniform in color, very saturated in color, very narrow in color, that depended right at the edge of the band gap of the lowest transition. And the quantum yield was about 20%, 15 to 20%. And I thought, oh my God, they're so bright. You can see them with your eye. And it was so much brighter than 0%, which is what I had started out with, in Bell Labs. And then Horst Weller, a colleague in Germany who was working on similar, he said, 20%, 15% means that at least 80% of your material is crap. And, and he was right. But, those sorts of comments are actually really important because they drive you to do better, right? They drive you to do better. We don't work in isolation. Today, the quantum yield is 100% for the commercial materials. And this is the graph from Chris's thesis here shows the control that this very simple process had on the size of the particles. So the difference in size between the sequential spectra that you see here as you march from something that looks bluish with a bluish tint with an absorption of 400 to a very, very dark red for the 15 nanometer particles on the very top. The difference in size between those things is just, tens to 20 atoms, really. They just added to it. So you have this exquisite control over the particle dimensions. So then that allowed us to do things that, were kind of accidental, but these things ended up crystallizing, as quantum dots, basically crystals of dots. And if you think of the dot as an artificial atom some ways, then you're creating a lattice of artificial atoms where you can control then the material. It doesn't have to be cadmium, it could be any other material with an energy that can go all the way from the UV into the infrared. And depending on how close they get together, they begin to talk to each other. And so we started dreaming about making new solids based on the quantum dots as the building block. And then you can think of mixing and matching different sizes together or different materials together to create binary lattices or ternary lattices. And there's a whole field now. Chris Murray at Penn is working is a leader on this, of mixing and matching materials to create these artificial solids and seeing if we can get novel properties out of them. And we could make them to be fairly large. And there's Sherry Kagan, another early student from my group, also at Penn, now married to Chris. And this is October 1988. Sasha Efros is carrying this little child here, who's actually the son of David Norris that's sitting there next to me. So Sasha came from Russia. He worked with Ekimov, and he had a party every year in October that he called the Red October Party. So we would all go there together. And these are my three students there's Manoj, David, and Sherry, and David's two sons, and Tim Harris from Bell Labs, and his wife. Okay, so then we started to do physics, which is really what I wanted to do. And if you go to low temperature, you can start to extract the real interesting physics of these harmonics, which are the excited states. And what you see here is a graph showing on your right side are the big particles, on your left side are the small particles. And you can see the harmonics sort of come together as the particle gets bigger. And what I was interested in was this idea of when do the bulk properties emerge from the atomic properties. This was what I had proposed, the curiosity part, right? And so for the small particles on your right, well, those are kind of like atomic properties. You can see these harmonics pretty well spaced together, and as the size gets bigger, they sort of get closer and closer to each other. But there's right around there, they start to deviate from that behavior. And as spectroscopists, we call that an avoided crossing. There are two transitions that want to cross. One flattens out, the blue one wants to go flat, but it's avoiding the other one. And as a spectroscopist or the physicist, you look at that and you say, wow, this tells me something really fundamental. And what it is telling you is that at this point, that's when that electron, that wave, is actually seeing that the box has atoms in it. It's not an empty box. It's a box full of atoms. And that electron and that atoms are form a periodic lattice. And that periodic lattice, now that electron scatters off that periodic lattice, and that is the spin-orbit coupling that you are seeing there, the spin-orbit coupling of that semiconductor that's appearing, which was not there at the smaller particles. This is a transition. This is one. So every property has a transition at a different size, right? And this is the size for that property. We also, so again, science is not, we're not isolated. So I went to ETH, I gave a talk at ETH Zurich, and I met Urs Wild, who was there. And then I also met W.E. Murner, who was at IBM at the time. And they were working on single molecule spectroscopy. Wow, this is really amazing. You can see single molecules, the fluorescence of single molecules. This is an amazing thing. And I thought, single molecules, they're all the same. So when you're doing single molecule spectroscopy, what you're really studying is the environment of the molecule. But for quantum dots, they're all different. So if you could do single molecule-like spectroscopy on quantum dots, we would be able to extract out the real properties of individual dots from the averaging of the ensemble, which is what we do when we have a gazillion dots together. They're all different. It washes everything out. And so we tried to do single molecule, single dot spectroscopy, and we're able to do that. So this is an image at room temperature then of looking at single dots in a microscope around 1996. We're still working on that today. And today we can then use these single dots to do really incredible physics with them as sources of light for quantum optics. And I think I'm going to skip that part because I'm running too late. So this is what you see at room temperature. You see that they actually blink. And the reason why they blink is because they're really nanosensors of their environment. They blink because they're charging and discharging, right? The little charge, there's a charge that hops on the dot that turns it off. And then you can think of building devices that are based on that, where you can turn on and off the light of the dots. You go to low temperature, you can see a spectrum. You see everything is much narrower, and you can start to see vibrational parts. So let me go ahead and skip this part, which is very complicated about using them as quantum optics building blocks, and go to the applications. So if you remember, in the 1990s, there was this amazing discovery from a guy in Nichiya, Nakamura, and he realized how to make gallium nitride as a blue emitter. When I started at MIT, and when I was at Bell Lab, there was this conference called 26 Semiconductors, the 26 conference, and everybody went to that conference and talked about zinc selenide, because zinc selenide was going to be the magical thing that was going to create a blue diode, right? We had red diodes, but we didn't have blue diodes, right? And zinc selenide worked great at liquid nitrogen temperature, but boy, they didn't work so well at room temperature. But they were, all over the world, people were working on zinc selenide, and Nakamura in 1990 figured out how to make gallium nitride a blue emitter and changed the world, really changed the world. And so, the spectrum of a quantum dot absorbs great in the blue at 465 nanometers, where that arrow is, unlike organic dye molecules. And so we immediately thought, as a community, well, you know, our material is really well suited to be coupled to gallium nitride diodes as an emitter to really multiplex things, because you could have things labeled with quantum dots of different sizes, use one of these new gallium nitride diodes, and start to keep track of different things based on the color or the size of the dot. And I even wrote a patent called Inventory Control, talking about, you know, encoding information with different size dots and color, and using a gallium nitride diode to read that information out. Still something that's really important. And that basically gave rise to the applications of quantum dots today. One of the first applications was in biology. In biology in 1998. Why biology? It turns out to be probably the hardest of the applications, because we were chemists. And biochemists are close to chemists. So we knew about proteins and things, you know, and stuff. And so myself, Paul at Berkeley, and Shuming Ni at Indiana University at IU, started thinking about labeling cells with dots. There was a Quantum Dot Corporation, was founded in 1998, co-founded by myself, Paul, and Shimon Weiss, who's at UCLA now. And this is a picture from it. And what we thought were really important was the fact that these dots were little pieces of rock. So unlike dye molecules, they would be much less prone to photobleaching. And you can see this is in picture. The dots are the red labels of the nucleus, and Alexa 488 is the green emitter here, and you can see the Alexa dye sort of fade away as the dots stay constant. So this picture came from Quantum Dot Corporation. I think it irked molecular probes so much that they decided to buy Quantum Dot Corporation to make it go away, right? So, but then Molecular Probes was bought by another company, which became Life Technologies, which is now part of Thermo Fisher. So this whole thing is now part of Thermo Fisher. Then I went and thought about going from cells to labeling in vivo through a chance meeting with an oncologist, and we had coffee together, and we started talking, and that started a collaboration in early 2000s, and eventually working with people at MGH. And so this is a kind of really hero experiment that we were doing, where in this case here we're looking at very rare cells in the bone marrow in these mouse models, and their interest, the interest of my collaborators, was to understand the microenvironment surrounding these cells, which are essentially like stem cells. But it needed to be a multicolor experiment and needed to be done in vivo. And the process for doing it before was to extract the stem cells out, label them, and put them back in. But the process of extracting them, labeling them, also caused them to become, to differentiate and change. And you couldn't do that if you were really interested in studying them. So, but you could do this with dots in vivo. And this is a 3-color experiment. You come in with red dots that are labeled with antibodies. And there's a few receptors that are overexpressed in these rare cells that you can then target. And then you can look for yellow. You look for yellow in this multi-photon microscope, and then you see a few of these rare cells, and you can start to study them in real time and watch them. This turns out to be a really hard experiment. We also went and realized that, unlike dye molecules, we could go into the near infrared, to what's called the SWIR region. So this is a mouse model of a brain tumor of a glioblastoma, and this is all labeled with dots. This is a time series now. The red is the first injection. So those are the arteries. You wait a little bit, you see the appearance of the veins, and if you wait even longer, everything fades out except the dots that are in the tumor, because the dynamics of flow in the tumor are much longer than in the blood vessels. Then if you now take composite particles that are made of indium arsenide that emit in the SWIR region around 1.3 nanometers or so, micron wavelight or so, and then you use night vision technologies, night vision cameras, you can see the individual particles. And on your right here is normal brain tissue. This is diseased brain tissue. And if you do the video, then you can see how fast the blood is flowing in the normal brain and how weird it flows in the tumor area. You can see that there's some areas there's no flow at all. The particles just go back and forth, right? And so that allows you to visualize directly why it's so hard to treat brain cancer. You just can't get therapeutics in there, right? It tells you why the tumor environment is hypoxic. You know, you build up carbon dioxide, why it's acidic, because the byproducts can't get out. And so their interest was to make this look like that. And so being able to visualize it directly is really interesting. But the real application, so this is all fun, right? But this is not at scale, right? This is great for research. The real application came in early 2000s, when for me, was meeting with an electrical engineer, Vladimir Bulovich, and he looked at these particles. He was part of the OLED crowd. So he had grown up with Universal Display Corporation and was interested in organic light emitting systems. And he looked at the fluorescence from these dots and thought, oh my God, I can use this for displays. And so we started working together. There were a couple companies that were started, Nanosys in 2002. I was part of that along with many other nanoscience people like Paul and Charlie Lieber and those, that crowd. And QD Vision eventually coming out of MIT. Both are now part of bigger companies. So Nanosys was now part of Shoei Chemicals as of a couple years ago. And QD Vision was taken by Samsung. The first application of the dots in lighting and displays was this light bulb. 2010. This was the birth of LED lighting. And when the LED light bulbs first came out, they were really expensive, like $100 a piece, right? And the color was not quite, really bad, actually. It was very blue light. There was too much blue light. And so by putting red dots on top, you could convert some of the blue into red and warm it up and have something that humans like to see, you know, with a lower color temperature, but still retaining the color quality that you needed. You could buy these things from bobs.com. I have some in my office still. Didn't last very long because the cost of buying an LED light bulb plummeted, you know, when China started making them at scale. It went from $100 to $10 a piece. And when you have that, then there's not enough profit to be made by incorporating the dots in them. And then you have to put them directly on the diode. And the diodes got hot, and the dots didn't survive the heat. It's still a challenge today, and their company is still working on that to understand how to make them less fragile. But learning from that, the application that came out was actually in displays, because you could use the same idea then to make RGB in a light guide, and that was what LCD really was about. And so the first application, the first product came out of Sony in 2012 using blue diodes, these incredible 465 emitting blue LEDs, and then converting some of the blue to red and green light through the quantum dots, and made 2012, 2013 made these QD-based televisions that contain cadmium cyanide, which really surprised me, because for 10 years before, every time Sony came to MIT to ask about what we were doing, they had a small list of elements that were forbidden. And cadmium was one of those elements. And, you know, but somehow they put cadmium in those TVs for 1 year. And they're over-engineered, and they work great, and I still have 2 of them, although it took me a couple years to get them. So I went to QD Vision. I said, you know, you should give me a TV. You know, and they said, well, you know, Sony won't even give us one. So I had to wait a couple years until they were on sale on Amazon, actually, to have them delivered. I still have them, and they still work. But Samsung saw that and decided this is a great technology, but indium cadmium cyanide was not going to be good for anything. So they figured out how to make indium phosphide dots at scale in large quantities, which we could not have done in academia. The cost and the labor to be able to create those materials was not something that we were ever going to be funded for. You really needed a big company with investing, $25 million or $50 million and believing this to be a technology to do that. And that is the QLED TVs. And so they bought QD Vision, and really what they bought was the logo. Because if you look at the logo, the Q has a little bar on it. If you take away the bar, it becomes an O, and then becomes OLED, right? And LG was selling OLED TVs, and now Samsung could say, we're better than LG, we sell QLED TVs. So that little bar was worth tens of millions of dollars. And then today, the way that they make the technology today for these quantum dot enhanced televisions is basically combining the two technologies together. You pixelate blue OLEDs, as you would generally, so you have one color OLED, and then you inkjet print on top the RGB, the red and green dots, and so you get the red and green from the dots and the blue from the OLED. And so the day of the Nobel Prize announcement, that same day I got an email from Samsung Display asking me where I wanted my new television. I thought, wow, this is great. So I told him, the biggest you have, and bring it and send it to my office. And that was a lot faster than Sony. Application in solar energy, in night vision. So this is a UV emitter, a UV diode that's attached to a secret briefcase here. This is a scene that my student and my collaborator from Raytheon put together. Maybe it takes place in 1960s in Berlin somewhere, and they're exchanging secret documents, and the Russian spy has a night vision camera and wants to keep track of the secret documents and puts a little blue LED, UV LED on one of the briefcase that contain it, and he has a special camera that has quantum dots on it that is able to see the UV. And so part of this part here is the dot enhanced night vision camera, and on the right side is without the dots. And you can see as Frank here stands up, basically what the dots are doing is taking the UV light and downshifting it into the infrared so that the camera can see it. And as he walks across, you can see that the normal camera just can't see UV at all. So it's very, very easy and very powerful. Flexible displays and PV. And so the field now has expanded way beyond semiconductors. The same idea of the synthesis and the characterization that we did back in the early 90s is applied to metals and semi-metals, magnetic materials, ferroelectrics, et cetera. And we've created, as a field, this new periodic table of nanomaterials that have all sorts of morphologies that we can combine with organic materials that now have real applications, displays, they're made at scale, biomedical imaging and sensing and solar energy, and the emerging technologies in electronics and optics and quantum systems. The heroes, are always the students. So if you multiply the number of students that I've had with all the groups that are doing this, there are really thousands and thousands of people now working in this field. So we came from being outsiders in the 1990, with me and Paul's group and Lewis's group at Bell Labs. They were the only people working on this in the US, to today having an incredible number, a big community. And I do want to say thank you to the sponsors, and also re-emphasize the need of fundamental research and funding for fundamental research. So, we have a really unique system in the US of inventing fundamental science, which is unparalleled anywhere in the world. And we're in the middle of destroying it. And it's really, really depressing to me, coming from France and Tunisia, with my father coming to the US, this great country, that we are destroying what is the economic engine of the US. When I came to MIT, so how does it work? Well, you hire assistant professors. They have great ideas. They are untested. Universities like Stanford and MIT and Harvard and these places use their own resources to give startup packages. millions of dollars to 28, 29, 30-year-olds to do curiosity-driven research or applications with very little except a vision, right? And we take a chance on these people. We take a chance on these people, and very often it's at the beginning of their careers that they come up with the new directions, the new discoveries, as I did. Everything I did at the beginning was funded by MIT, by my startup package. I didn't have any preliminary results. I couldn't have gone to funding agencies and say, give me money, I have no idea what I'm doing, but please give me some money. That was not going to work, right? That wasn't going to work. It's not until I had preliminary results that I was then able to go to the NSF. Also, the role of foundations at the beginning was incredibly important, right? So it was MIT money and then foundation money. The Packard Foundation was an incredible resource for me, the Sloan Foundation, the Dreyfus Foundation. All of these were super important at the very, very beginning when it was just an idea and a vision. Eventually, you get some preliminary results. NSF steps in and starts to help you with money. I had Presidential Young Investigator Award, which is now the Career Award, was super important. The DOE has been fundamental to my research from the beginning. The support of DOE has been incredibly important to my research this whole time. The NIH, the Army, et cetera, all funded a lot of the fundamental stuff. And then when you have applications, then the companies step in. The Samsungs, the DuPonts, the Siemens, et cetera. But it's an ecosystem and it's a timeline that is really unique to this country. It's nowhere else in the world. And if we get rid of curiosity-driven research like this, then we don't have the pipeline for 20 years from now. Where are we going to get the discoveries of today that are going to be changing the world in 20 or 30 years? Why is it that most of the Nobel Prizes are from the US? There's a really good reason for that. It's our ecosystem. So in any case, thank you for this, and I'll take your questions.
Fantastic to see you give about 4 decades of colloidal quantum dot history. It's from fundamental all the way to applications. Certainly, I want to look a little bit longer time scale of, let's say, few thousand years. Material science has been so important. In material science department, we talk about material is a label civilization, right? Now with the modern materials, quantum dot is one of the very exciting ones. The past 40 years lay down the foundation. Any prediction for the next 40 years?
No. So one of the things I learned is, humans are terrible at predicting more than 10 years ahead of time. We're okay at predicting 3 to 5 years. 10 years is really hard, and 20 years is impossible. So I'm not going to go there. But it's going to be wonderful. It's going to be wonderful. I'm sure it is.
Very nice and inspiring talk. You mentioned about the curiosity-driven research and how it's under attack. But there was also the Bell Labs mode of research, which you played a part in. So do you have any comments on lack of Bell Labs now? Which does material because transistors.
Yeah, Bell Labs was a really important part of history, of scientific history. And it was really special, and there's nothing like Bell Labs today. It was really special because it really brought a lot of different science together. And AT&T was a monopoly. It's never going to happen again, probably, because AT&T was a monopoly, and it was a monopoly that was running the long-distance network, right? The phone system. And it didn't really care so much about what Bell Labs was doing. As long as you were doing research on communication, very broadly defined, then you were probably doing something useful. Bell Labs did a lot of things for communication that AT&T benefited from, like the fiber optic networks, the lasers that drive communication, et cetera. All that came out of Bell Labs. But so, and the funding, the way that it was funded, when it was still a monopoly, researchers would write a paragraph and say, I have this idea, and bring that to their department heads and make a pitch for it, and yeah, that was the proposal, right? And there was a lot of equipment, a lot of money, and part of the secret of Bell Labs is that the managers knew that the people in the trenches, the scientists in the trenches, had a better idea of what to work on and so it was really driven bottom up. The research was driven bottom up with a lot of leeway given to the scientists there.
Please join me in thanking our speaker again. I'd now like to invite to the stage our inspiration for this event, and one of the role models who helped introduce me to the pillars of the department, that striving for excellence while always maintaining that sense of collegiality for the whole community and every individual in the community. Professor Nix, our Lee Otterson Emeritus Professor of Engineering.
Well, thank you, Sarah. I know that I am what separates you from lunch. So I'll try to be brief, though I do want to thank a few people. Thanks, Sarah, and also thanks to the MSc faculty and Alberto, particularly for setting this whole thing up several years ago and putting it on. I really appreciate it. It's been really great fun, I think, for all of us to meet once a year and renew our friendships. I should also then thank Chris and Lisa, who probably did most of the heavy lifting in getting this organized. Now, as I've said before, and I'll say again, all I need to do to know that the Material Science Department is in good shape is to listen to Jennifer and Colin and any of the other MSc faculty about when they talk about their work. That just shows where we're going, and the ship of state is sailing very well, as you could see yourself. Now, it was a real treat for me to meet again Stephen for coming here and giving your lecture. He probably had to suffer through some of my classes. I'm sure he did. But I must say, though, that he may be the only person who remembers that the pyramids are a much better way to track how dislocations move in face-centered cubic lattices. You and I may be the only people that remember that. But no one else says, what's he talking about? What's he talking about? I know, and it was terrific. And now, so he was our alumni speaker today. And speaking of alumni speakers, most of you know that 2 years ago, Wally Rhines, one of our distinguished alumni, was our speaker. And some of you know this, but I would like to report that 2 days ago, Wally was elected to the National Academy of Engineering, which is a very big deal. So, Wally, you're here somewhere. Where are you? Are you? He left. He still deserves a hand. And it was, if you've already heard, it was really a special treat for me to meet for the first time Moungi, and for him to come here and give his brilliant lecture. So thank you, Moungi. And I think I can speak for everyone here to say that you could teach in my department anytime you want. And as some of you also know, the same 2 days ago, Moungi was elected to the National Academy of Engineering. So congratulations. And as you've already heard a couple of times, my grandson, Oliver, is one of he's a co-advisor of Oliver at MIT in chemistry. Now, I'll be finished here pretty quickly. Now, many of you know that the MSc department has been my academic home for a very long time. Some of you may know that I came here as a graduate student in January of 1959. That was 67 years ago. And this has been my academic home those nearly 7 decades. Now, in that time, I was really fortunate to develop what I have been calling my academic family. And a few of those folks are here today, and I want to recognize a few of them. And I'll start with mentioning 2 of my earliest students. And what I want them to do is to stand and then remain standing as I introduce other students. So Bill Coughlin, Bill, was my 6th PhD student. He graduated in 1969, so that was a while ago. And then also from about that time period, David Matlock, who was my 13th PhD student, graduated in 1972. So our connections run back a long time. Now, I could also, I've recognized Bill and David. I'd like to ask what I call my extended academic family to stand. Here I mean my own PhD students and let's say people with whom I've published, may not have been my formal students. Could you also stand? A few of you are here. Please stand. There are some here. You have a few. Good. Please, don't be bashful. And if you published with me or I with you, actually more I with you. You wrote and I got my paper. And E qualifies. Thank you. But please standing. And then maybe just to go one step further, anyone who took courses from me, there may be okay, now, all right, now we know. So, yeah, okay, there you go. Thank you all, and you may be now seated. Now, it occurred to me that Bob Sinclair, are you still here? Yeah, Bob Sinclair might be thinking that this was something like the scene of the old British movie, Goodbye, Mr. Chips. You remember that old movie? Anyway, this old doddering professor and his students show up, and anyway, that's a joke between me and Bob. I'll just end then by thanking my family also. Today represented by my son, Jeff, and his wife, Kat. Where are you? Somewhere? There they are. Please stand, both, yeah. And the infamous Oliver, who is also somewhere here. Where is Oliver? There he is. And so thank all of you for coming and making this really another memorable event.
Great, thank you. So that brings us to the end of our public speeches. I want to remind everyone that during the lunch hour, we have posters directly across the hallway. And new for this year, we had one of our alumni suggest, Brian Harden, I don't know where Brian is, suggest that to facilitate increased interactions between our students and our alumni, we should get alumni to volunteer to represent their companies and to sit at the tables across over where the posters are. And we had a number of our alumni volunteer to do that. And I'd like to personally thank you for that. Our students and postdocs are so excited to meet with you. To help with crowd control, I'd like to both recognize our poster presenters and those alumni who are going to volunteer by asking them to stand so we can all clap for you. And then I would like to suggest that those individuals might go to the front of the lunch line. And the rest of us, you can look at posters if you want to wait for the crowd to die down. So with that, thank you all so much for this wonderful day. It's an honor to have been the MC for today. Thank you and enjoy the rest of your afternoon.